No México tá quieto.
Eu
quero ir.
Você é um tranqueiro, cara.
Quero almoçar lá perto da Rocinha, lá tem um restaurante muito bom que eles falam lá.
Perto da Rocinha, perto daquele hotel lá,
Inspiration. The chair of this section is Professor João Batista Calisto from CIEMP, Brazil. So Professor Calisto, thank you very much for organizing. Professor Calisto is going to introduce the speakers of this session. So please.
Okay. Good morning, everyone. Welcome to this section that is a very, very important section for Brazil. It's timely because biodiversity today is a global problem, important for Brazil and for the planet. And I think that you have a very nice section because you have four speakers, two from academia, university, and two from companies, pharmaceutical companies or startup companies. So I have the pleasure to introduce the first speaker. This is Dr. Cristiano Guimarães. Dr. Guimarães has received his PhD in theoretical organic chemistry from Federal University of Rio de Janeiro in 2001, under the supervision for Dr. Ricardo Bica. Then Dr. Guimarães worked for many times in America, especially as a postdoc student at the Yale University first, and then he moved for Amgen, and then for Pfizer, both as a senior principal investigator. So Dr. Guimarães has very experience in drug discovery, both in chemistry, but also in biotechnology. And he started the Brazilian company, startup, Nintex. But before this, he moved to Brazil and worked for many years in a shared laboratory. This is the big one, pharmaceutical company in Brazil, when he was the innovation director. And then he is now the CSD and co-founder of Nintex biotechnology company. Dr. Guimarães has a number of publications and is also very cited, and is included 2% of international scientists of the world more cited according to Elsevier. So I have the pleasure to introduce my friend and collaborator, Dr. Guimarães, for the presentation today.
Thank you. Thank you so much. So it's a pleasure to be here. So I would like to first congratulate DNDi and LOLA for its 10 years. So it's such a noble project and work that you conduct. So thank you so much for the invitation. And today I'm going to talk a little bit about Nintex at the end of my talk. But before that, I would like to, show some slides. Waiting for it to. So I know that the tree is very beautiful, so that's why it's stuck in the first slide. But while we wait, oops, sorry, this is dangerous. This is like full of traps, right? So you have this and... Just a second. Sure. Like in Brazil, right? So basically, I would like to share with you some ideas in terms of how we have an opportunity to be a player and a relevant player in the global scenario of R&D. First, because the business model today is decentralized. So the R&D in pharma is done collectively. So you have startups that and biotechs that do basically the early stage work. Okay, thank you. And you have the big pharma, which basically develops these compounds. So, okay. So that's why I was saying the current business model in pharma R&D, it provides a big opportunity for Brazil to be inserted in this R&D, global R&D, and to be a relevant player in pharma innovation. So I'm sorry about the busy slide here, but basically what we show is that because of the patent cliff, the model became decentralized, right? So the companies had to rely on external innovation basically to be productive. And you see the number of projects from 2001 to 2019 in global pipeline of programs grew from basically 6,000 to I think 19,000. And it's interesting to see that in this plot here, 20% of these projects are from companies that have only 1 or 2 projects. And 50% of these companies in this pipeline here have 1 or 2 projects. So basically, you have a lot of companies that are working early stage, developing these programs, hoping that at some point they will license out to big pharma. I'm going to describe a little bit of these graphs for you, showing that because of this change in strategy, now you see more and more often small biotechs registering compounds or registering drugs. You see that in 2009, the top 10 pharma accounted for 31% of approvals, while biotechs accounted for the same 31%. But in 2018, the numbers were 16% and 63%. And this graph here shows that from another paper, that 65% of the compounds FDA approved were filed by the top 20 biopharma companies were externally developed. So again, I mean, you see that big pharma doesn't do. When I was at Pfizer and Charlie too, remember that the model was, we do everything internally, silently. We develop our compounds, and then we hope that they're going to be registered. But that's no longer the model. So as you see, because of this increasing number of transactions, there are basically 2 gates where you see the licensing happening. First is the preclinical gate, and the second one is phase 2. And it's easy to understand that because, at the end of the preclinical, you have POC in animals, and at the end of phase 2, you have, safety and POC in humans. Another thing that is interesting to see is that the attrition rate in the preclinical stage is 65%. So basically 1 in 3 projects, they move to the licensing target zone. They take basically $5 to $7 million to develop and 5 to 7 years. It's interesting to think that when you think about Brazilian pharma companies which are focusing generics, that's not very different from the time and the money that you need to develop a more complex generic molecule. But the upside is that the valuation of these programs, even in the preclinical stage, are huge. I mean, you're talking about, this is present value. This is not necessarily how much that program is going to bring in terms of revenues, but you see that obviously the valuation goes up as you reduce risk and you advance your program. But if you license out a compound here in the preclinical stage, it can bring roughly several, from tens to few $100 million at the end of preclinical, and at the end of phase 2, this number is basically from several $100 million to $1 billion. So it's interesting because now you can talk to Brazilian businessmen and, the private sector and say, this is not so high risk and long term. Now you have a strategy that fits in the pocket and fits in the mindset of people who have money here in the country, so which are somewhat risk averse and want fast returns. So, and it's interesting, and we're basically already exercising this and seeing examples in the country where this is happening. The first example is from Recepta Biopharma. This company is run by the ex-president of FAPESP, Professor José Fernando Pérez. His son is president of the board of directors and also is the managing partner of Fundo Pitanga, which is an investor of Nintex. They did the out-license of this monoclonal antibody in preclinical stage in 2015. And at Ache, the founders of Nintex and Atlas, who is here in the audience, and the rest of the team, we implemented this model and we started, developing compounds, extracts in the preclinical stage, and we are successful. I mean, we did that 3 times already from 2017 to 2023. So I have a vitiligo, a natural extract from the Brazilian Biodiversity for Vitiligo, which is in phase 2 now. It was licensed out in 2017. And we have 2 small molecules from Ache's drug discovery, which these are not necessarily out-licensing deals, but they are partnership deals or co-development deals with Big Pharma. In this case, it's Big Pharma. McQuaid Center is from Otsuka, which is a leader in the central nervous system therapeutic area. So we out-licensed this in 2021. This is small molecule for PTSD. And after I left Ache, I know Atlas, who is running the ship there now, so beautifully concluded this transaction. So this is happening. I mean, the pharma companies and I would say Ache, but I know that there are other examples in Brazil. They are trying to develop compounds early stage. And at least Ache's model, and which is also Nintex model, is doing that in preclinical so that we can have the budget and the time so that we can monetize the company. So, but I showed you one example of a monoclonal antibody, one that is a natural extract. The other 2 were small molecules. But if you think about a platform so that Brazil can be a relevant player in the globe in terms of R&D, in pharma R&D, we have to think about the Brazilian uniquenesses that we have here. And obviously you all have to cite, that we are the most biologically diverse country in the world. We all know here that natural products are highly bioactive. They have evolved to have a function. So, and since we share genes with plants and microorganisms, it's obvious that molecules that work for these living beings, they can be good starting points for us. Not only you have a rich biodiversity, but we also have a rich traditional knowledge in terms of the use of medicinal plants, vibrant science, and this is more what's happening in the globe, that advancements in omics, data analytics, artificial intelligence and analytical technologies, which are now turning natural product-based drug discovery more tractable. We will hear that not only in this session, but in the afternoon. We have a partnership with CNPEM, with CIEMP, and we have what some consider the most advanced biodiversity law for access and benefit sharing in the world. So that was a problem in the past, but now is something that catalyzes research using our genetic heritage. And natural products and the Brazilian biodiversity are perfect. Already 5 minutes? 5 minutes of questions as well? Oh, all right. So it's a challenge, right? So the challenge is that we as scientists, we tend to simplify problems, right? So we have been successful in pharmaceutical field, simplifying problems and diseases. The magic bullet approach, which one molecule has to reach one target is somewhat successful. I wouldn't say somewhat, but is successful. But there are a lot of patients who don't respond to treatments because a lot of the diseases are multifactorial. You know, I would say most of them, they are a combination of not only genetic factors, but environmental factors, and most importantly, how these two factors interact with each other, especially through the microbiome, the gut microbiome. So a lot of diseases are linked to the gut microbiome. Not only diseases that are intestinal bowel disease, or but you have like cancer, you know, patients who don't respond to immunotherapy because they are in the state of dysbiosis. So we have even like neurodegenerative diseases that are now linked to the gut microbiome, and it's easy to understand because we are 10% human. 90% of our cells are microbes. If you talk about genes, we are 1%. So it's we are a complex individual, not only from the human cells point of view, but we are basically controlled by these microorganisms who live inside of us. So we need to then think about, if you want to do a reemergence of drug discovery based on natural products, we must consider the full range of their biological effects, not only the reductionist approach that one molecule, one natural compound against one target, and it's not surprisingly, if this study was conducted by Professor Rob Knight at UCSD, it's called the American Gut Project, and you see that what modulates the gut microbiome the most is the consumption of plants, So now I have an opportunity to use natural products to modulate not only targets directly, but modulate targets indirectly via modulation of the gut microbiome. And it's not only modulating microorganisms, but when you think about the intestine, it's not only epithelial tissue. You have neurons, you have immune cells, you have enteroendocrine cells. It's basically the molecules don't even have to be absorbed. They modulate you in situ, in the intestine. It's interesting that there are experiments that you increase GABA in the intestine, the GABA receptors in the brain, the animals go up. The GABA molecule doesn't get in. It's through the enteric nervous system. So, and it's very easy to understand. If you see the concept about the One Health concept, the soil is the great repository of microorganisms, and we all get these bugs from the soil. The animals, the plants, and us. So obviously, since the plants have been interacting with the microbes in nature for millions of years, what modulates our bugs the most is the plants. And now we have the Brazilian biodiversity here, which is not only a repository of molecules to modulate targets directly, but to modulate microorganisms. That's why we created Nintex. I don't think I will have time to go over this, maybe just a few slides about the founders of Nintex. As I said, we used to work at Ache. I was in research, Miller in development, and Saverio in business development. This is our strategy, to use natural products to modulate targets directly and indirectly. Have a platform where you use artificial intelligence to structure this traditional knowledge, the literature on natural products. We have like very good partners like CNPEN and CMP and Centroflora along this platform to develop these programs. We have our proprietary technologies. We have a simulator of the gut microbiome, so this technology is called XGI Biomics, and the first reactor simulates the stomach, the second the intestine, and the last three, the three portions of the column where we inoculate the fecal samples. And we can understand how, and can screen extracts, we can screen natural products and understand how they can modulate the microbiome. And we have a pipeline of projects with our partners. I forgot to mention that we have been invested by Fundo Pitanga, by Mr. Guilherme Leal, who is one of the owners of Natura, and Mr. Peter Anderson, who is the owner of Centroflora. That brings $3 million to the company in the seed stage. Also, we have money that comes from our partners, like Embrapi funds and money from the companies who partner with us. which bring another $3 million. So we are well funded to conduct this pipeline. And we hope to inspire others to think that in Brazil, we have a good model, we have a good platform, and we need more companies to do the same thing as Nintex is proposing to do. Thank you.
Thank you, Dr. Guimarães, for the presentation. An important initiative in Brazil for startup in biodiversity. It's important, especially for microbiome interests, that is very, very important today. And science is more important, June of the world. Every day we have some news about microbiome and disease. Now I would like to invite the second speaker, Professor Dr. Monica Rupp. Dr. Pupo, is full professor from University of São Paulo, Ribeirão Preto. Dr. Pupo works in School of Pharmacy of University of São Paulo in Ribeirão Preto, where she is currently holds the position of Deputy Director. She is a natural product chemistry, whose researcher has focused in understanding the role of specialized metabolites in interspecies interaction in plant, insect, and microbiomes also. It also is the discovered natural product for infectious disease. She earned her PhD in chemistry from Federal University of São Carlos in 1997. She was a visiting scholar for John Clark Group in Harvard Medical School in 2006 and 2007. She has coordinated international joint grants, including the International Cooperative Biodiversity Fund supported by FAPESP and CNPq. So I would like to call Dr. Monica.
Good morning. Thank you, Callisto, for the introduction. Thanks, Jardel and Luis, for the invitation. It's a pleasure to be here. I also congratulate LOLA for the 10 years. And let's talk a little bit more about microbiomes in the academia context. So here I highlight what my laboratory is interested in. So we are looking for ecological interactions in hotspots of biodiversity in Brazil. We apply new methods for microorganisms isolation, and we apply all methods in natural product chemistry, from the most traditional ones, bio-guided fractionation and isolation and the structural elucidation, but guided by modern methods such as metabolomics, genome approaches, and big data and bioinformatics. The bioassays are carried out by collaborators, especially in the CEPID CB4 that's coordinated by Glaucius Oliva. And by doing all these, we aim to discover new biologically active natural products. So why microbiomes? We already heard about that. So microorganisms are everywhere, and they establish interactions with all organisms. We have been studying microbiomes of medicinal plants, amphibians, insects, and also soil microbiomes. We try to understand the functions of natural products produced by the microorganisms in such complex interactions. And we believe that having this chemical ecology guide, we can more rationally discover or identify hits, especially for infectious diseases. So today I'm going to talk specifically about some examples. Insect microbiomes. Insects are the most diverse group of living organisms on Earth, and they establish different interactions with different microorganisms. The benefits exchanged include dispersal, nutrition, and protection. We are interested specifically here in the protection because bacterial symbionts of insects produce metabolites that help insects to defend themselves against pathogens. Oops. Sorry. Better now? Okay. So one example here that highlights a big study that we did in collaboration with other people. We sampled a lot of streptomyces. Streptomyces is the most prolific actinobacteria genus in the production of bioactive natural products. And here we can see that streptomyces from insects, they are more active against fungal and bacterial pathogens when compared to streptomyces from soil and plants. So one specific group of insects we are interested are the social insects. They farm fungi for food. So this is a very ancient example of agriculture, and they include the termites in Africa and the ants, fungus-growing ants and stingless bees that are native to the neotropics, and we have a lot of species in Brazil. So basically, we can illustrate this symbiosis in this picture. The insects farm fungi in mutualistic interactions, but they are not the only players in the systems. There are other microbes here. As those colonies are very populated, a lot of individuals, they are susceptible to specific diseases that are caused by bacteria and fungi. And one way during evolution that insects acquire to defend themselves from those pathogens is by the association with bacteria in the phylum Actinobacteria that produce selective antimicrobial natural products. So those molecules can kill the pathogen and have no effect on the fungal food source. So it's a very, this system evolved millions of years ago, so we can learn from that. And here are some molecules already identified, new molecules identified in different insect symbiont systems. In our pipeline, we run this, both of these approaches, the chemical ecology, so we want to understand the role of the molecules in such interactions, and also here we try to identify hits for medicinal chemistry. And here is very important to focus on new chemical entities. So as part of two projects that we are and we're involved, here I show specifically in the antiprotozoal context, we had this number of extracts from bacterial symbionts of insects in our lab, screened against Leishmania, Donovani, and T. cruzi, and we got very decent hit rates. So we selected some of those extracts to pursue the chemical studies. And here I show this strain Streptomyces 311 from Siphomimex ant. It was active against the specific ant pathogen and also against human pathogens. The metabolomics showed it could produce new chemistry. So we did all the work and ended up isolating and identifying Siphomimesis. That's a new polyketide. I can move here. Okay. And Siphomimesis was active in vitro against fungal human pathogens that have drug resistance phenotype. And most importantly, it was active in vivo models of candidiasis. And in addition, Siphomimesis and two related analogs were also active against leishmania, intracellular leishmania, amastigotes with very good selective indexes. Other examples of bioactive compounds from streptomyces, from ants, include those molecules. They were already known in the literature. However, the antiprotozoal activity had not been reported. And here I highlight the two polyethers here, also with good activity and good selectivity. More recently, from multiple strains of pseudonocardia, that is an important symbiont of those fungus-growing ants, it's an actinobacteria. We isolated and identified this new non-ribosomal peptide, named the actinomycin, that is important for the ants to defend themselves against the specific fungal pathogen. And actinomycin also was active in the in vivo model of candidiasis. As I mentioned, we are also looking for symbionts of stingless bees that comprehend an important group of native insects in Brazil. They are important for pollination of native plants and also for agricultural crops. And here I show the activity of some actinobacteria against the honeybee pathogen, that's Penibacillus larvae, a bacteria. All those extracts of those actinobacteria, they showed also antiprotozoal activity. We identified different classes of compounds, including new ones that are shown here in blue. And here I highlighted two of them, meliponamycins A and B. They are analogs. Those compounds were active against the honeybee pathogen and also showed activity against leishmania. So showing that we can align this chemical ecology approach to natural product discovery. So to finish, I haven't seen the 5 minutes alert, so I can probably talk more. To conclude, I hope I have shown you that natural products, we all know that natural products have a successful record in drug discovery and development, especially in the field of anti-infectives and antibiotics and antifungals. As we heard from Cristiano, natural products have evolved to mediate interactions between organisms. They have not evolved to be drugs for humans. This is an anthropogenic vision. So we need to learn from nature. So symbiotic systems are very interesting systems to prospect new compounds. So we can definitely align this chemical ecology to the medicinal chemistry. We also know that Brazil has untapped genetic resources to be studied. And we also heard that new technologies have improved natural products discovery. So we have opportunities for basic science, also for innovation, and also important to establish public policies to the preservation of biodiversity. So thank you very much. I also thank my wonderful collaborators and people in my lab, former students and current students, and also the funding agents. Thank you very much.
Thank you very much, Dr. Pupo, for a very nice speak about natural product chemistry and possibility to use in drug discovery. Now, I would like to call the third speaker is Dr. Atlas Azevedo, That is Dr. Azevedo is a R&D director, head of drug discovery and external innovation at Shea Laboratory. Dr. Azevedo is a biomedical scientist with extensive experience in pharmacology, industry, leader of drug discovery, R&D program, and external innovation initiatives. With over 13 years of experience in the field, Dr. Atlas is currently employed at Shea Laboratory, serving as R&D director and head of drug discovery and external innovation. Dr. Atlas has a PhD degree in genomics and bioinformatics from the University of São Paulo and master in business administration from FIA, I suppose in USP, São Paulo. Please, Dr. Azevedo.
So I'd like to thank the organizers for inviting Shea and myself to be here today. It's really great to see the academic efforts that have been done in this area of the natural products, and it's been a really pleasure to witness the foundation of one of the first startups in this area as well in Brazil, which was well funded, formed by scientists that have both experience in the academic and also industry. So we've been seeing a lot of movement in Brazil in natural products, and also the interest in accelerating that and taking advantage of all these powerful resources that we have here. So I'll just say a few slides about the efforts that Shea has been doing in this area as well. For those that don't know, Shea is one of the largest pharmaceutical companies in Brazil. It's a national company with annual revenues of around $1 billion. We've been recognized as the most innovative pharma company for the past 10 years. Well, we have basically two technology platforms for drug discovery, one that is focused on small molecules, and the second one that's focused on natural products. We've named, in fact, when Chris was there also with us at Shea, we've named this platform as BioProspera, which is a platform that is focused not only on technology, but also on establishing the network of partnerships that will be required for doing natural product drug discovery as well. I'll try not to be repetitive here, because I won't teach the priest about that, but why natural products? Because it's definitely a very refreshing and relationship between humans and natural resources. In Brazil in particular, which is the next slide, I think is the most important one. Brazil not only has one of the largest natural biodiversities in the world, but a lot of traditional knowledge on the use of those drugs as well. So I think this is a very important aspect to mention, that we are not starting from the scratch, right? So there is a lot of knowledge on the traditional use of those plants that can be accelerated to make new drugs as well. Brazil also has a very rich social biodiversity. Basically, everybody has some sort of connection with the natural world in Brazil. It's a very cultural thing, and that can be also engaged in R&D. Is one of the most biologically diverse places, well-developed legal and regulatory framework, as Chris has already mentioned. Natural products have been really a prolific starting point for new drug discovery as well. And a growing number of natural product research papers have been published internationally by Brazilian scientists. And it's interesting to see the level of the research that's been doing. I think it was really interesting to see the work that Monica has shared. So we are really reaching a novel plateau on R&D in this area as well. And we established this platform trying to discover and develop innovative medicines for natural sources, including the Brazilian biodiversity. Something important to mention is that we are not focusing only in the Brazilian biodiversity, because we know that there's also opportunities in other natural resources as well, but we are trying to, as we are being in Brazil, right? So we really want to accelerate the powerfulness of our biodiversity in this area as well. We are combining cutting-edge technology, technical excellence, and the largest vegetable biodiversity. And it's in fact a statement of a strategy that believes that the excellence of the Brazilian innovation can gain market differentiation and put the pharmaceutical industry, and I would say that all the Brazilian scientists in the global market of science, right? Science is also we can also generate economic value in the pharma industry using science as well. So we have two different models that we are exploring. The first one is what we call bioprospection. We are doing a lot of work with the CNPAN. We have Daniela here and Marjorie as well. We are collaborating with them for several years already, and the idea is to collect libraries from 5 biomes and run established compound libraries and run those against, especially using target-based screening approaches. Not exactly that we're now focused on phenotypic, but we believe that there are good targets to tackle that will be really a challenge to tackle with a pure small molecule, and therefore we have a lot of potential on using more chemically complex natural products as starting points for drug discovery purposes. We also are working in the ethnopharmacology field, so basically trying to prove or make sure that traditional knowledge or basically the pharmacology that is being published in the literature can be translated into new medicine. So it's very interesting to see how many publications Brazil has in this area as well. Lots of natural extracts, isolated compounds from the nature have been published, have published results in pharmacology, in vitro and in vivo by Brazilian scientists. And so we are, here there's an important aspect that we're doing is that we are integrating a lot of this biomedical science knowledge, and we established, in fact, so before that, just mention that we are using state-of-the-art technology to enable drug discovery in natural products, even collaborating with a collaboration with PhytoBios and CNPAN, not only for generating the chemical extracts and isolating compounds, but also for screening those compounds as well, and using X-ray and generating crystallographic structures for SBDD approaches as well. It's a very interesting interaction between those players, because basically everyone has a different expertise that we can put knowledge together and make sure that we're moving from the screening up to the preclinical and clinical development, which is something that Shea has already experienced doing. And we know it's been a great interaction with those groups. We have a couple of projects, and we expect to share more information about them in the future as the projects evolve. As I was saying about ethnopharmacology, I think an important aspect to mention is that there's a lot of available information that can be integrated using AI. But not only AI, basically by capturing, structuring that information and make that information available to scientists, there is a lot of hypothesis generation that can be done. So we've established an in silico prediction and research lab inside of a shed that we call Inspire. This lab has had one of these first projects, the structuring of a platform that we call Bioprospera DB. So it's a proprietary database that basically integrates in a very automated way information about natural extracts, compounds, isolated from them, targets, diseases. So we have this available, and it's really an asset that the company generated to make hypothesis generation in a very fast-paced way. And now what we're doing is to use machine learning and prediction methods to generate new hypotheses as well. So, because there are some of those hypotheses that basically finding patterns as a scientist, you can start already projects, but there's a lot of untapped potential on using machine learning and AI approaches to do so as well. We have inside of a molecular design and synthesis lab, and it's interesting that in the past 4 years, we also started establishing the resources for the lab's infrastructure to isolate natural compounds as well. So we're doing this. So we have both the capacity to isolate natural compounds and also to synthesize them organically by organic chemistry in general. We have internal expertise working in a very highly regulated environment. Brazil has a very tough regulatory framework for natural products. In some cases, especially for CMC patterns, even more challenging than Europe, for instance. So this creates also an opportunity to make really very standardized natural extracts and isolated compounds from them as new drugs. I think Cristiano has already mentioned about AS012, which is a natural extract for vitiligo treatment that is currently under phase 2 trials, both in India and in the US. It was partnered with one of the global players in the dermatology field. We have Ashifla, which Professor Callisto has had a really prominent participation on the discovery and development work that was done by Ashe in the past, and it's definitely a success case because it was the first topical anti-inflammatory, was the first natural extract that was developed as a drug in a pharma grade in the last years. This drug has been commercialized in more than 15 countries as well. So it's a very interesting example on how innovation can generate exporting revenue also for Brazil and put Brazil on the global at the global scale for pharma in the pharma field. Last but not least, as Cristiano mentioned, drug discovery is a risky approach, so you've got to have a pipeline. It's a funnel of innovation, right? So we know that most of our programs will fail. So we currently have 8 projects at least. This is a little bit outdated. We have a couple of other projects that were included in the pipeline. And we expect that some of them will reach the exceeding points of preclinical, phase 2, and even the regulatory approval in the future. It's a very diverse mix of natural extracts, but also of isolated compounds. And depending on the disease, competitive field as well, we are defining what is the best strategy. Natural extracts have some advantages in terms of multimodal mechanism of action, but have a lot of challenges whenever you think on oral administration, think on the pharmacokinetics of those compounds that, as Monica mentioned, they were not developed or nature didn't create them to be drugs. So they don't behave as drugs pharmacokinetically wise. So depending on the disease and the mechanism of action and the route of administration, we are making movements toward isolating those compounds as well or working with the natural extracts. That's it. Thank you so much for having me today, and be happy to answer any questions you have.
Thank you very much, Dr. Hatila, for a very nice presentation, and I can witness the effort of Axé since the last century, interested in natural product. I know the owner of the Axé, this time he loved natural product. So I think this Axé laboratory is one of the most important lab in Brazil, pharmaceutical company that invests in research and development. So I think that is important, especially now in terms of biodiversity. So it is very nice. I think it is very important for Brazil and for scientists that we can do something. Okay. Now, I would like to introduce the last speaker this section. Is Dr. João Roberto Losset. Dr. Losset is Senior Discovery Manager for DNDi. Dr. Losset is a pharmacist by training, with a PhD in plant chemistry from University of Lausanne, before completed postdoc fellowship in London School of Hygiene in tropical medicine. Dr. Losset earned a diploma in public health and tropical medicine from Humboldt University in Berlin. Prior to joining the DNDi, Dr. Losset acted as scientific collaborator and lecturer in the School of Pharmacy of the University of Geneva and the University of Lausanne, and where he has academic research focused in discovery of new antiprotozoal drug from plants, and included the supervisor of several PhD student, master, and undergraduate scientists. Dr. Losset joined the DNDi in 2005 as a consultant, and today as a collaborator. Thank you, Dr. Losset.
Bon dia and good morning, everyone. I'll take the safe version, as my voice is already half gone, so I'll try to make it as clear as possible, taking a safer risk. So it's a real pleasure to be with you today. I think that I don't have to say much more about the importance of biodiversity and research in natural products to you in Brazil. I think the former speakers actually did it quite well. Coming from a tiny country, Switzerland, with a beautiful but limited flora, I think Brazil has it all. And I think this is really the spot where you could really engage into natural product the discovery research. The only thing I want to underline is that I was really impressed by the talk yesterday from Prof. Elena Nader, who really insisted that nature should be treated with respect and not be seen as a commodity that you can consume without any consideration. It's an heritage that we should transmit to the next generation. So I think this is quite an important statement, I think. Without further ado, what I want to discuss and go through today is to give you a bit of a breast of what we've been doing at DNDi in terms of sourcing of new lead series for neglected diseases, and the current status and the challenging we're facing, hence the role of natural product, with a bit of rationale, not talking too much about it because this has already been highlighted previously. The challenge is then to work with natural products. I'm a natural product chemist myself, and I had often hard times in the lab to deal with them, actually. So I think this is something that should not be underestimated, and I'll try to convince it that it's something that has to be taken seriously. Then I'll go through a few examples, so where I see and foresee some innovative technologies that could be really helping, if not game-changing ones, to address natural products research and discovery of new lead series out of natural products. So working backwards at DNDi, I've been involved with DNDi since 2005, where we started with low throughput assays based on basically intracellular assays with limited capacity to evaluate those compounds, few hundreds per month, that was maximum. And I think that a breakthrough came at the time where a team at Institut Pasteur Korea, under Brazilian leadership, it's quite important to be said, with Professor Freitas, Lucio Freitas, developed the high content, high throughput assay systems against intracellular form of Leishmania and Krusei. On top of it, obviously developed several different assays, but I'll stick to those two here for my presentation. And this allowed it actually to start screening in a big way via different approaches, but really to process thousands, tens of thousands of compounds a week. And additional groups, University of Dundee, GSK, Novartis, and others have screened massively, and that led to quite a massive number of compounds that have been screened so far. I think we can estimate 5 to 7 million against Leishmania and Krusei for each of the two diseases, with quite a lot that has been done over the last, let's say, 10 years. We focus very much on classical chemistry, meaning compound coming from synthetic origin, and haven't really worked on any natural product during these 2 decades. And as we've developed also chemoinformatic tools to monitor the progression in terms of, selection of compound libraries, but also the redundancy related to the chemical series we identify from those screens, we saw really that quite a number of the hits and the series started to get redundant. And I think this is a concern because the chemical space and the number of collections existing is limited. Hence, we need to look for other types of sourcings to address attrition and to address redundancy. I'm going to go fairly quickly through this one because this is something that has already been discussed. Natural product has been the template for a number of drugs that are currently on the market. They are biologically pertinent. They've not been explored much against neglected diseases. And also they produce high hit rate related most likely to this pertinence in biology against biological targets. And this has been nicely documented in a paper written by Novartis and published. And I'm happy to share all this reference with you, obviously, or discuss this after the talk. Some of the challenges associated with natural products is really that there's a lot of traps. There's a lot of things that you should be aware of, starting from high attrition, ubiquitous compounds, pan-assay interference compounds, very well described by Jonathan Bales in a specific version focused on natural products, on paints, catechol, quinone, and other types of compounds that recurrently hit or different assays, but still continue to get heavily published by a number of groups. So, and also novelty, and this is some paper, it's quite small probably here, but a publication by Roger Lennington's group, who has showed basically looking at time auto distance, that also that groups publish and discover new compounds. A lot of this novelty based on time auto distance is actually not real novelty. It's more plateauing or even decreasing over time. So I think this is a concern. Other things that needs to be taken into consideration is the time and resource it takes to go through the classical route of bio-guided fractionation, isolation of the product. Obviously, that probably quite a lot of you are familiar with the loss of activity and other things that prevent you from identifying something promising. So how can we address those things? And this comes to a few examples I want to show. The first one is mostly related to pure natural product. This is the first thing we did as DNDi when we engaged into natural product. We went first to look for the landscape of new collections as pure compounds and try to identify opportunities of library, well characterized, chemically speaking, to see if we could purchase, acquire some of them and screen them against our bugs. And this is a collaboration that we've performed with Sandexis and GARDP, identify those libraries and develop actually chemical tools to triage and to evaluate and to prioritize those collections. So without going into the details on how we've done this, we develop Bayesian models that are capturing quite a number of features, not relying on one single physical property like sp3 or log-d, but having a more holistic approach and develops through different type of algorithm, fingerprints, based on extended connectivity of fragments, fingerprints, try to put some categorization, what we call natural product likeness, as those collections were coming from natural product isolated from plants or from microbes, but also a number of those collections were natural product inspired. So we wanted just to rate them and evaluate them and develop this type of these models to help and to assist. So they were part of a larger type of evaluation, natural product likeness, but also other type of consideration, how those compounds would add in terms of new clusters to what we had screened in the past, and other consideration. We try not to put too much emphasis on druggability, Lipinski-like rules, as they're not applicable to natural products, but we also to consider a number of other things, as I mentioned before, like ubiquity of compounds that I think we won't be able to push forward. Obviously, cost and other conditions to access the collection was also taken. So we purchased actually 4 collections out of this and identified a number of series that we pushed up to a certain point, but has to drop eventually. Those are the few examples. I'm not going to the details for sake of time, but we faced some issues related to metabolic stability and to mechanism of actions that were suboptimal to address the disease we were interested in here. So another example, but this is really an example now, not moving to mixtures and how to deconvolute efficiently and quickly mixtures of natural product, is a piece of work based on molecular networking that we work through with my former group with University of Geneva and Pierre Favre. So for the sake of time, I don't want to go through the detail of it, but it's basically you take information from MS2 spectra that you compile and center on one single sample. You bin basically the ion and neutral loss of all the peaks that you can elute and record, and you vectorize them, and then you compare the vector once you line them, and based on this information, you can cluster and project this into dimensional views and make sense of those molecular network with the entire molecular information based on MS2 then, that you can really look more globally and align this with different layers of information, should it be taxonomy or bioactivity. And this is something we've done in collaboration with Pierre Favre, with data from Swiss TPH40 Cruzy, where we've located a number of clusters related to interesting networks based on this mass spectral information. And via interrogation of database using text mining, identify a number of compounds and pathways, genetic pathways. And one of them that was quite interesting was actually rotenoids that were not really being described previously as being active against CRUSI. This is CRUSI data I forgot to mention. And then we went for direct isolation of those compounds and were able to confirm nanomolar activity of a number of rotenoids with good selectivity. You might say that rotenoids as such won't be able to be developed as drugs, but the interesting piece for us is to demonstrate this workflow is actually working, is quite efficient, and also maybe to fish mechanism of action out of series that can be identified through this piece of work. Another thing is that most of us, we screen in a monoparametric way, looking for activity and exclusively activity, with the risk of being redundant and identify molecules attractive, but maybe not really related to mechanism of action that you want to investigate. The interest of imaging that you can bring into this is that you can extract eventually other type of information following compound perturbation of your SS system to assist and classify mechanism of action via the features you can extract from imaging, and then take this into account on the top of activity to isolate your compounds. This is work that has been pioneered by Anne Carpenter's lab at MIT, and I'm not going to detail this more here, but this is a nice piece of work done on mammalian cell lines that we're currently applying with IPK, with Institut Pasteur Korea, on T-CRUSI intracellular assay using a set of dyes and extracting with really suitable equipment proper to this type of work. So laser HSCS equipment, 6,000 features that we are reducing in terms of data. It's obviously quite a bit of data work and analysis to get this information into a clusterization and to look at this from a mechanism of action standpoint, using or not using reference compounds. So this work is ongoing. We adapted the assays. We already evaluated a set of first compounds. The workflow is functional, and also the cluster analysis, multiple cluster analyses are functional, but obviously now the next interesting step will be to see where this data will lead us. Last but not least, I just want to mention a number of tools that do exist, and this is really also a recent progress that is quite important and relevant for those who are involved in natural product, with a number of data depository, structure, information, NMR, spectra, MS, spectra tools that can allow you to better manage and organize your data, etc. So, and there's a need obviously to better connect all those tools and to make them speak across to get more data out of more information of those. But this is really excellent tools for open research. So to conclude, natural products are a unique source for new leads. I hope that I've convinced you about this. There are a number of things that you should be conscious of working with natural products. It's not easy work, and there's a number of traps that you should be avoiding. There are game-changing, innovative technology that will really put you in a better position with more information to select the material you should focus your efforts on. At the end of the day, you still need to isolate and characterize compound one way or another. And I think that there's more and more tools and more groups who are publishing, releasing data, posting data in databases and in information systems that are going to help the scientific community to work more efficiently on natural products. I think I'll stop here for sake of time. Thank you.
you, Doctor. Was that very nice presentation, overview of natural product in the rock discovery. So I think that you have time for some question, comments. And I invited the speaker to sit here in the table and you open for discussion and comment for.
So should I start? Okay. So Peter Sjö from DNDi. I'm just asking, maybe Monica especially. So I really love the kind of see the structures, everything, but as maybe everyone noticed, they are quite kind of complex structures. So I was thinking, what is your kind of next step in the strategy to start to investigate, modify these compounds? Do you have access to kind of large-scale fermentation capacity and purification, etc.? What is your strategy? I'm a bit interested how you approach this.
Okay. I don't have capacity in my lab for large-scale fermentation. We can do multiple rounds of fermentation in order to get more compounds. But of course, this is possible to do, but for this we need the collaboration. And people who are expert in translating this lab scale fermentation to a higher, because it's different. The microbes, they work differently in Erlenmeyer flasks or fermenters. So, but it's possible. To me, for now, what we want is finding the compounds and if they are of interest, so we need collaboration to go further. But we also needed to investigate the mechanism of actions of some of those compounds, because they were just active in phenotypic assays. So this is another thing that must be done.
Hello, I'm Srini here from Novartis. Excellent presentations, really remarkable achievements over the last. Excellent presentation, really remarkable presentations from all the four of you. The question what I had is, has there been any improvement in production of these natural products to a scalable level where we can be used for treatment of things? The second question I had is, topically these molecules are available for to be active on the target of interest, but oral bioavailability has been a challenge for natural products. Has there been any research in this direction to make these natural compounds orally bioavailable so that it can reach either Chagas or any other systemic diseases?
Thanks, Srini, for the questions. I think there's no easy answer to your questions. I think it's case by case. I think and step by step. Production is really based on what you find and how you can optimize the compounds. Is there, for instance, example of Taxol, is there a precursor that is easily tractable and maybe that you can modify with one last step of synthesis and get really hands-on massive amounts of material and avoiding to chop up the bark but use the middle instead, for instance. That was an example to make it sustainable, for instance. And there's a number of processes related to bio fermentation or modification of the enzymatic pathway to increase production of products. As artemisinin and others, I think they've done a number of tentatives to also obviously synthesize it de novo as well, and to see whether you could compete with the demand. So I think it's really case by case, depending on the complexity of the structure and the yield that is produced in the natural in the microorganism or in the plant. So then I forgot your second question.
Thank you, fascinating talks. I love natural products. I'm a natural product chemist at heart as well, so great. Another question for Monica Pupo, actually. Specifically for cyphomycin, it's really quite interesting activity against leishmania. Do you have any insights yet on mechanism of action or plans to do that? Would you be interested in some help to do that? And any plans to try and take the compound forward as an interesting candidate?
Yeah, we actually, we had a project together with DNDi, University of Dundee, to carry out some experiments to try to figure out the mechanism of action, but we didn't succeed yet. So yeah, unfortunately, but yeah.
Which suggests it might be a new mechanism, because they're very good at identifying common mechanisms.
Yeah.
Thank you so much for your talks. I learned a lot. And I have, in fact, a question that I would like to hear your opinion about it. And considering that we are here celebrate the 10 years of LOLA and all the, and even the goals of the discovery of new drugs, I'm wondering that, in fact, we are not here in order to find new hits or lead. Our main goal is find a drug candidate. And in fact, when I see most of the structures, they are completely far from the idea of a drug candidate. So my question is, why you can converge the idea of anti-parasitic compound that should be oral administered, not be expensive, with the compound that you see here. How difficult could be the lead optimization process? And what do you think about it? How is possible to do the optimization of these compounds?
They're dodging the question and like sending our way. So this is more of a question for you, but what I have to say is, I mean, if you think classically, I'm gonna say in general terms, okay? So if you think classically, right, you always seek for oral absorption and plasma exposure and distribution and finding a target. But since I'm a new fan of the microbiome, I think a lot of the effects, right, of these compounds are not via the classical, pharmacology, medicinal chemistry way of thinking. Most of these drugs or these molecules in nature, they evolved to have a physiological function in the plant species, but also to interact with the microbes. So I think a new compartment that we have to consider is how these natural products reach the colon. So forget about absorption. They don't need to be absorbed. They don't need to be absorbed. I'm not talking about infections, okay? I'm talking about in general terms. And modulate the microbiome. They can be metabolized by the microbiome. The microbes have an enzymatic capacity that rivals the liver. They're kind of complementary. The liver oxidates and conjugates. The microbiome reduces and deconjugates. So, and there you think, oh, okay, and then they're going to be absorbed in the colon. They don't need to be absorbed in the colon once they are metabolized. They might change the fermentation of those microbes. They might change the composition and the components on the wall of those microbes. And there they interact with the cells, not epithelial cells. They interact with neurons in the gut. They interact with enteroendocrine cells in the gut. They interact with immune cells in the gut. Another thing that is very important is that this interaction for multifactorial diseases between environmental factors and genetic factors happen because of a syndrome called the leaky gut syndrome, right? You have the tight junctions because of inflammation. They are destroyed. A lot of things get in. They are not supposed to get in. And this, for example, in natural products, you have polysaccharides and or the elements on the surface of the microbes. They can induce the repair of these tight junctions, and they can basically avoid this interaction between the environmental factors and the genetic susceptibility. So you have to pay attention and kind of like take a step back and consider this as a new way of treating diseases. So when you do that, things will get easier in terms of drug-like properties, but it will be more difficult in terms of understanding the mechanisms because it becomes much more complex.
Can I? Yeah, this is the question for the Ashe presentation. There was a comment regarding of the Brazilian authority for approval of products. My expertise is just with the FDA, where it is very, very stringent regarding the specifications for the allowed impurities of drug substance, drug product. Could you elaborate a little bit about the Brazilian requirements for that?
Yeah, sure. I think my comment was more related to the European legislation, which tends to be a little bit flexible depending on the country. But we have experience discussing with the FDA this project for natural extracts, and it's very interesting because they tend to regulate a natural extract depending on the point of view as a biologic or as a small molecule, depending on how you see it, the CMC requirements. So for instance, they do have a very strict control on the internal standard of this extract, but in the meantime, they can ask you about bioassay for quality purposes, for instance. So I think that is really dependent on the case, right? Whenever you have a natural extract, usually you know a lot about its chemical composition. So you know if it's a particular class of compounds that has an effect, or if a particular isolated compound would have the majority of the effect and should be controlled. But definitely developing a companion bioassay for quality purposes, I think would be required for any FDA purposes. But in Europe, I think they are more flexible on trying to understand the effects and how you are controlling over the several manufacturing batches that you're doing. So that's it in a nutshell. Yeah.
Hi, Daniela from Sandy just a quick comment on natural product production and bottlenecks for heat to lead optimization, as commented before. Just remember that most of the drugs, historically, 70%, I think, are not expired by natural products, but 6 to 20% are the natural products themselves. So if you can find good natural product heats, they are complex to be produced and so on, but you can be inspired in these structures, especially if you can decipher the mechanism of action and then simplify the compounds or use the target to develop a pure synthetic molecules that are very trackable in industry, right? So there are few examples nowadays of these that have been very successful, especially in the current era that we are now with artificial intelligence, to put structural biology and so on. So I think this is a traditional bottleneck of natural products to obtain the molecule, but now you can easier use the information and then enlarge these bottlenecks and be successful in natural product research, drug discovery, you know? Just that. I don't know if some of you want to comment, but just
okay.
Not best.
Some comments here. No.
A quick comment on what Daniela has just mentioned. It's interesting that in our collaboration we see this project in which we have 3 different series, and one of them is exactly like that. So one of the series is naturally inspired. So we identified a new allosteric site, and then we did the screen by natural products, and then we did a screening and found a chemical series that is chemically tractable, etc. Having said that, I think that the post-pandemic situation has taught us that developing national supply chains will be really interesting. And since we do not have like all the organic intermediates that will be required for organic synthesis, why not developing drugs that would come from natural sources in which we could manufacture the natural extracts and isolated the compounds here in Brazil. Apart from all the green chemistry associated with that, which will be quite cool, that's also this independence that could be helpful for the country, because we won't have the same development in terms of chemistry that we are seeing for East Europe, China, India. That's off, right? So it will be a way of developing our national supply chain as well.
Klaus.
Yes, following that question, perhaps for you and Cristiano, how do you deal with the IP problem of being unable to, protect natural products, at least from the Brazilian point of view?
Yeah, I would say, Glossus, there are ways of, circumventing these. There are like IP strategies that you don't necessarily have to patent the extract, but you can patent a process. You can, patent maybe a modification that your process, induces in the molecule, like a transformation that only your process does. There are like extraction techniques that privilege a specific compound that in nature exists in smaller concentrations. So in IP, there's a lot of storytelling, right? And how you convince the patent during the patent examination process. One thing that is interesting is that if your chemical entity is extract, that in itself is a protection, because it's very difficult to reproduce. You have to plant that, you have to cultivate. There are like a good agricultural practice that you have to use, and then you have your own process, and then you have to generate your own data. This CMC to develop it, I would say, is more difficult than a biologic. And then for others to copy, there are no like generics for example, plant extracts. They have to start all over from scratch, basically. So that in itself is a big technological barrier, more so, I would say, than IP.
Now you have some questions sent by internet. So I asked to.
Yeah. So actually received lots of questions. It's very good, and please send us more questions. And we select some like from the Vinicius Bonato, who would like to ask for Cristiano. Vinicius says, I would like to know your opinion on the role of computational chemistry tools in studying biodiversity to discover new compounds. It's a majority of research using artificial intelligence methods, or there is still room for physics-based methods, since I believe most of the assays are phenotypic or related assays.
Yeah, I used to be a computational chemist. It's been a long time, but I would say, like Atlas mentioned, we basically developed the Inspire. He named the lab Inspire, but we had the idea that these had to be treated by artificial intelligence and data analytics and these tools, because all these databases they don't communicate with each other. So you have some databases with plant extracts and compounds, others with compounds and activity, others with targets and diseases, others with compounds and microbes. So basically we said that, and they are tabulated. So it's very hard to make sense when you look at them and see if there is a potential compound in our plant extract. So There are like tools we use in our case at Nintex, a tool called Knowledge Graph, where you basically now can combine all this information and it's very visual. You basically can filter based on the quality of the paper, the number of citations, the other properties in terms of activity against a target. So in this way, for example, in the project with Professor Callisto, CMP, we use our methodology to do a virtual screening of plants that had good connections with a specific disease. And we use those suggestions. We went back, we saw the papers. These tools make non-obvious connections because this information is not necessarily in a single database. So if you look at a database, you won't see necessarily the connection between a plant and disease. You have to put them together. And then it's very interesting that we made few extracts, and now you're running the animal models, and some of the suggestions were very active in the animal model, even so you don't have a publication between the plant and the disease. So that's where your IP can come from, because it's not in the literature. But the data analytics made that connection. So the physics-based part of the question is that, like Daniela mentioned, as you find a hit, and then you have an allosteric pocket. We are seeing a lot of allosteric pockets in a kinase. There is a project that Nintex has with CNPen, and it's a kinase, and we have like 4 allosteric pockets, and those plant fractions, they are functional. They inhibit the kinase, but they don't. The crystal structure doesn't show.
Sure. We have more one question, please. Very quick.
Sure. Doesn't show. I mean, it's a complex question, so it's a complex answer, but it's a physics-based answer. But then, I mean, you can move to physics-based after you find the hit, a crystal structure, and then you can play with computational methodologies.
More one question for internet.
So from Rafael Limeiro, he would like to ask for Jean Robert. Those millions of compounds screened against T. cruzi and Leishmania would be great value for researchers in neglected diseases. Do you plan to publish this data?
So it's a great question. So we progressed a number of those up to a certain point. We developed some of them, and some of these compounds and series are currently in lead optimization, etc. And we published part of this data. What we could not is to publish them all. It's a matter of time, it's resources, and it's a matter of prioritization. So I think, yes, we try to do our best to do this. It's part of our policy and our DNA to share data and to share information. We posted, for instance, a number of data packages in a publicly available database such as KMBL, for instance. And we're happy to share data and to discuss sharing of data. But obviously, to publish it all, it's a bit of it's a task on its own, and we need much more resources to do this. Obviously, there's a component that is also related to the contractual relationship we have with a number of our partners as well, with respect to confidentiality and data dissemination. Not that it's something that should prevent us from publishing, but it's obviously something that needs to be addressed before publication.
Okay, I have my own question, but more a comment, because I know that answer does not exist, so as a challenge, but it's necessary. Drug discovery from natural product in pure molecule is not easy. It's not easy, it's quite difficult, and chance for not success is so big, so big. If students, some people like, read the history of Taxol. Taxol demands 25 years after the discovery to put in clinic. And because it's a molecule quite important, too much complex, and the synthesis involved 25 steps, as published in Nature many years ago. There is another question, another problem related to mechanism. It's improbable that plant compound has just one mechanism of action. It's quite improbable. Majority of plant compound enter to the cell and involve many, many targets. The Chinese scientists is now recently publishing very most paper showing that 20, 30, 35, 40 different targets for just one molecule. The problem is nobody knows the pharmacokinetic to demonstrate where the effect occur for each different disease. And also how the compounds cause off-target effect and the same doses.
Well, I don't think that's the session we are presenting now. That's not me, but anyway. When I was first invited to coordinate this section, I felt privileged. Privileged to be here, to learn from you guys, to share my experience. But today, with everything that is going on in the world, in particular, the conflict between Israel and Palestinians, I feel even more privileged. Privileged to be here, safe, in peace, to be able to discuss good quality science with you guys. Science that can change the world, can improve the quality of the life of the most neglected ones, that can help improve us as society, because we definitely need to improve as society, as human beings. So today I feel honored, happy, and proud to introduce the outstanding speakers of this section. The first speaker is Professor Elder Nakaya. Professor Elder is a molecular biologist and has extensive training in bioinformatics. An expert in systems in immunology, he works at the intersection of system-wide measurements, networks, and predictive modeling, particularly as they related to vaccines and infectious disease. His lab concentrates on investigating the foundations of infectious disease through computational system biology. Dr. Nakaya serves as an adjunct professor at Emory University School of Medicine and the Department of Pathology. And he also shared with us he's passionate about science, soccer, teaching, travel, and according to him, gorgonzola cheese. So welcome, Elder.
Thank you so much, the organizers. And I have to update the cheese. Actually, my favorite now is burrata, but I forgot to. But so today I would like to talk about harnessing network medicine to discover potential drug targets. And I know that some of you are not familiar with the concept of network medicine. So that's what I want to start. So if you take a group of people that are infected with any disease or treated by the same drug or vaccinated with the same vaccine, you're gonna see that actually they have different outcomes. Some will be okay or asymptomatic, some will have like a severe adverse events or is gonna be very sick, and some will even die. And that happens because although they have the same perturbation, they are completely different, they are very unique in terms of immune system. They have different molecular factors, stress levels, malnutrition, gender, age, everything influences the outcome. So when you measure all the biological components of these people and create a network connecting these different biological components, it's gonna be like a hairball. It's very hard to analyze. So the job in my lab is try to find which small part actually of this network is better associated with the outcomes. Of course, by taking into account the the covariates. So I would like to show some examples of how we do this and how this can be actually used for revealing potential drug targets. So I'm going to start with Zika, which it's a major effort of several researchers. And we got brains of babies that die of right after birth, and they had congenital Zika syndrome. And we did transcriptomics, proteomics, and genomics. And I'm going to skip a lot of the results showing just the integration of all these biological components. And of course, we try to validate some of these findings to try to understand why only some of the babies are going to be born with this syndrome, whereas others not. But when I was presenting these results in another meeting, one of the professors of UFMG, Mauro Teixeira, he was surprised when he saw glutamatergic synapses. So this sounds new, is what people from Minas used to say. So he came to talk to me, and then he was surprised because what he was investigating is that actually that glutamate could be related to the congenital Zika syndrome, because glutamate, in addition of being amino acid, is also a neurotransmitter. When it's in excess, it can be neurotoxic. So the idea was that maybe Zika is somehow dysregulating the glutamate pathway and leading to neurotoxicity. So what he was trying to show is that maybe if you use an antagonist of the receptor, you can prevent the neurotoxicity. So they set up an experiment. Actually, Vivian, I think, is going to be one of the speakers. She did an infection with primary neurons and Zika and showed that the neurons die and the virus replicates. So when you put the memantine, this drug which is safe to give to pregnant women, it actually prevents the death of the neurons, but the virus still replicates. So it shows that potential leads that this could be maybe somehow used in the future to help pregnant women. So Jonathan in my lab, he was interested in COVID, as many of us. But the problem is that there was already over 50 transcriptomic studies of COVID. But if you take 2 of these studies and then you compare the list of differentially expressed genes provided by the authors, saying that, okay, these are the genes that are important to COVID, you're going to see actually that the overlap between these lists is very little, meaning that the consistency of the findings is not strong. So what he did was to analyze dozens of studies in the same way with tissues and single cell data. So we have, for example, not only 1 blood study, but 8 actually blood studies. And then you can define robust signatures, for example, of 206 genes that were reliably differentially expressed in severe COVID. But then we also look at different organs, and then we know the genes that are consistently changing in severe COVID in different organs. So when you put everything together, when you combine the genes and the organs, and then you see that some genes are actually connected to several organs. And one of the genes that was there was this RAB8B, which nobody knew anything about this gene in the context of SARS-CoV-2, but the RAB family is a family that is involved in several different viruses because it has to do with the entry of the virus and the exit of the virus. So what we did was a collaboration with someone in Unicamp, Pedro, that got human cells and then used CRISPR to delete the RAB8B. When you try to infect the cells, what we saw is that actually, if you have RAB8B, you have these viral loads. But if you don't have, the viral load decrease, meaning that the virus do need this protein to replicate. And of course, if you guys want to check your favorite genes, you don't have to reanalyze the data because we create a website where you can just type the name of the gene that you want, and then it's going to show which organ it is affected. So and now I would like to change to knowledge network, which don't have to do with the molecular parts. So we have a former president, which was a great scientist. So he said that actually chloroquine can be used to treat COVID. And he knows that because he investigated the molecular mechanism behind this. And how does he do this? He goes to PubMed, for example, and then type chloroquine and then finds a paper about chloroquine. And then in the title, he can see that chloroquine inhibits NALP3. And then he puts in the notebook, chloroquine inhibits NALP3. And then go to the abstract and then see that chloroquine prohibits the expression of IL-1 beta and IL-18. And then you put there in the notebook. And then you say, okay, now I'm gonna move to the next paper. But just chloroquine alone has over 28,000 papers. So it is impossible to do this exercise for all the drugs, for reading all the papers. And that has to do with the fact that we are publishing too many papers. So since 2000, we published 500,000 papers per year in PubMed. And then in the last 2 years, we published 1.5 million papers. So regardless of your field of expertise, you are facing a knowledge tsunami. And of course, no human can read 45 million papers, but a machine can do this. So that's exactly what we did. We asked the machine to read everything and then connect the dots, connect genes, drugs, and disease. And then we as a human scientist, can look at this network and then find out which insights we can take and validate in experiments. So we publish also the evolution of knowledge in different diseases. And then we create, just for fun, this graph that shows the number of papers published per year in Brazil, for example, in the last 30 years. But we did also for the whole world. But that is interesting to see that 30 years ago, we were publishing about schistosomosis, Chagas, but then our interests changed a little bit. I hope you guys can see the name of the disease there. If you guys want to cheer up for your disease, that's the time. Like, go, go, go. So look what happens in 2016. Zika. And look what happened. Guess what happened actually in 2020? Yes. So we went from infectious neglected disease to then disease that happens in the rich countries. And of course, as everyone else, COVID's in the end. We also use this information to do drug repositioning. So the way we did this, before of that, because we connect everything, we can see the relationship of the disease, in this case, neurological and psychiatric disease. We can also see the molecular mechanisms that are shared by different diseases, but we can also use for drug repositioning. So let's take, for example, one network, hypothetical network, where we know what are the genes that are related to two different diseases. And then if there is a drug that is used to treat disease 1, and you also know that the drug targets this protein there, and because you have the network, indirectly the drug also affects these other proteins, which are all shared by disease number 2. So maybe you can reposition the same drug to treat the disease 2. So that's the idea. And when we do this for psychological and neurological disorder, we found several drugs that you can never see a paper in PubMed showing that someone is trying to treat Alzheimer's, for example. But if you go to clinicaltrials.gov, or if you go to the internet, you're going to see actually that there are cohorts, clinical trials ongoing exactly to treat this drug, using this drug to treat the disease. So that was a validation that the method works to identify potential targets that are someone else is trying. We did the same approach for all human virus. So of course you guys cannot see, but each dot is a different virus. So that's the amount of information we know about HIV, hepatitis, and all the other viruses. And then, of course, we can combine this information for all these different groups of virus. And then we see that the respiratory virus cluster together, the arbovirus cluster together. And that's nothing to do with just the molecular part. This is taken from the literature. So when you see the pathways, you see that it kind of makes sense, the reason of some of these viruses are connected. We did the same approach for chronic inflammatory disease, Crohn's, lupus, arthritis. And now, of course, we are trying to cross-reference with the information of clinicaltrials.gov to see how much of this is actually already being tested and which ones are potential new targets. So this is just an example of some of the candidates that we found for some of the inflammatory disease. And as a conclusion, the precision medicine that we are all aiming to do is very different than the current medicine, because we now try to understand the mechanism of the disease. And because of that, we can do the treatment and diagnose based on this mechanism, and classify the disease based on molecular aspects. And then we can provide the individualized treatment, prospectively treat people, and early detect the disease. For these three parts, this understanding the mechanism and then provides the information, that requires network medicine, because if you do not have the full context, if you do not integrate information, not only the clinical part, the genes, proteins, metabolites, it's hard for you to understand the real mechanism that is behind when you induce the change of one protein, what's going to happen in the rest of the pathway, and etc. So with that, I would like to acknowledge my team in University of São Paulo and Einstein, and of course, you guys for listening. Thank you. She told you that I like soccer, right? So
Thank you, Elder. It was fantastic. Well, the following speaker is my dearest friend, Professor Carolina Horta Andrade, Associate Professor at Federal University of Goiás. She is a professor of medicinal chemistry at the Faculty of Pharmacy, and the head of the Laboratory for Molecular Modeling and Drug Design. She obtained her PhD in Drugs and Medicines from the University of São Paulo. Her research focuses on computer-assisted and artificial intelligence-oriented drug design, as well as on the development of artificial intelligence tools and QSAR models for toxicological research and risk assessment. She currently serves as associate editor for ACS Omega and is a member of the editorial advisory boards for the Journal of Medicinal Chemistry and the Journal of Chemical Information and Modeling. Carolina didn't tell me what is her favorite cheese, but I know she likes rosé wine, and we actually had a lot of wine yesterday, so I hope she does well. Welcome.
So thank you, Christy, for this kind introduction. And yes, we had too much wine yesterday, so hopefully I can deliver my talk now. Let's see. So first of all, I'd like to thank the organizers and the invitation, Luis, Jadel, and Luisa. I'm so honored to be here today and talk a little bit, just a flavor, right? 15 minutes of what I've been doing through these 13 years that I've been professor at the Federal University of Goiás. We are also part from this hub, which is Excellence Center of Artificial Intelligence, and my institution that is an Embrapa unit in artificial intelligence. And today I'm going to share some stories of applying artificial intelligence for hit discovery for neglected and emerging diseases. So first of all, thank you, Elder, for introducing the artificial intelligence and where do we use artificial intelligence nowadays. You know that almost pretty much everything we do, there is artificial intelligence connected on what we are doing, but how about in drug discovery process? So we can use artificial intelligence in pretty much all steps of drug discovery process, from target discovery to the post-market surveillance, even the clinical trials. So in my research as a medicinal chemist, computational medicinal chemist, we use artificial intelligence for lead hit and lead discovery and optimization, and also for ADME and toxicity predictions or properties. So basically, we are going to talk about more about drug discovery process and generative design. So we need data for the computers, right? So we say that we are living in the big data era. Regarding chemical and biological space, there is estimated that there are 10 to 60 chemicals discovered or described in the world. And from those, 10 to 33 chemicals that are drug-like chemicals that has physical chemical properties that are like drugs. So how can we mine all those compounds? How can we choose? How can we pick the ones that could be the right drug for the right disease, for the disease you are studying? So we can use or we can advance the knowledge that has been already generated and are in publicly available databases such as Campbell, PubChem, and so on, and apply the artificial intelligence models or methods to learn with the data that has already been generated. And in this case, we can use many methodologies, and one of them is what we call QSAR, quantitative structure activity relationships. Basically, what we do is try to correlate chemical structures with a biological property, each or any biological property that you have the data. So first of all, we need the data of chemical compounds and biological property experimentally measured. Then we are trying to correlate the variation in the structure, in the chemical structure with the biological property, generating those equations. And nowadays we use artificial intelligence such as machine learning and deep learning algorithms to generate those equations. We validate the model, and these models can be binary or classification model, or like active and inactive, or toxic and non-toxic, or the model can predict the real biological activity or the calculated biological activity. And how useful are those models? So we can build models for any data you have, biological activity, for example, and then if a good model is, oops, this is dangerous, yeah. So if a good model is developed, you can use the model to filter out some huge database that you have in hands of compounds that you want to try to test, to evaluate. So you can predict the activity of the compounds and select only the predicted hits, and then you are going to validate that. So we use those models to prioritize the compounds for the experimental validation, or to deprioritize the compounds that are predicted, for instance, as toxic or with low pharmacokinetic profile. So let's go to some examples. When I put my title of my talk, I wasn't aware that it was 15 minutes, so I will try to go very fast here. And I'm not showing any example for neglected disease, so we don't have time. I choose for emerging viral disease 2 examples. So I will start with Zika. So we started this project with Dr. Sean Atkins. He's from a startup from a company in the US. We started this in the beginning of 2016, and it was a collaboration project with a company, IBM Computers, and we used the supercomputing power of volunteers that are connected in a network that IBM provides. And with that, our main goal was to virtually screen all targets from Zika virus with all available chemical space in the world. So more than 30 million compounds in zinc at that time with the 10 targets from Zika virus. We had many papers in this project, and I chose to show you some results of this. It's a huge collaborative project with many researchers from Brazil. Professor Glaucius is here, Jair also is here from California, and this work has been developed by Melina, a postdoc in my lab, and Bruna, who is also here. She was doing her master's in this field, and we perform a parallel virtual screening for all Zika virus proteins. In this case, we used polymerase, protease, and helicase with more than one site for docking. We started with docking, then machine learning models, and then at the end we selected the virtual hits. We prioritized the virtual hits for the experimental validation in cell-based assays in the enzymatic assays. What we got, so for the protease, we got this hit. I cannot see the number, but I think it's something like 7 IC50, right? Yeah, and it's an allostatic non-competitivity inhibitor of the Zika virus protease. And also for the polymerase, we had many hits here, and the best one is here with 0.6 micromolar of IC50 in the polymerase. And I thought I had one more slide in this, but let me just go back. One of the compounds had this one, had activity in both polymerase, protease, and also showed some cytoprotective effect against the infected cells. Okay, so this is, in our opinion, a very good hit to start a hit lead optimization. Then this Open Zika project ended in the beginning of 2020, and we are finishing the virus project in my lab, and then it started the pandemics. So Sean called me, hey Carol, we need to write at least a perspective or a review. It's like a deja vu. How can we stimulate a drug discovery, an open science drug discovery project for a pandemic that is starting? So we published this paper in the early 2020, like in April, and it was a perspective, but we ended up with idea of a drug repurposing workflow, and then later we applied for some grants in Brazil. We approved some grants here in Brazil, and then we approved a big grant involving the BRICS countries with Brazil, South Africa, and Russia. And Richard is here because of this grant, so we are collaborating since then with Kelly Shibali group. So the idea is to start with the machine learning, deep learning models, docking, computational drug discovery in my lab. And also we have the collaborative effort of Sean as well in this project. And then we screened many different synthetic and natural products libraries and commercial libraries. And with the selected hits, we started the experimental validation with the targets. For this project, we selected 4 different targets, 3 from the virus, so the 2 protease and the spike protein, and also a human enzyme involved in biosynthesis, pyrimidine DHODH, which is also essential for the viral replication. With the hits validated, then we go for the, then we go for the structural studies, then the phenotypic validation of cell-based assays with Thiago Moreno from Fiocruz, who is also here, I think so, and cytotoxicity assays, and this is the cycle of hit lead optimization, right? So I will jump for the example. We have many good stories in this project, as it was a massive collaborative project with more 8 institutions. And this example I will show you briefly regarding the human enzyme DHODH. So this is a enzyme involved essential in the de novo synthesis of pyrimidines that are necessary for the viral replication. So if we inhibit the enzyme, we can inhibit the viral replication, right? So we develop many computational models, shape-based model, ensemble docking, as we had many data for this protein, as it is also a target for cancer, for other diseases. So there are many available data for ligands, inhibitors, and structural data. So we used all the data that have been already generated. We developed and validated the computational models, and in this case, we used the H3D database of synthetic compounds, and we selected 22 compounds that have been shipped to Brazil and then tested against the enzyme at Christie's lab. And we had this heat with 2.9 micromolar of activity, and we also confirmed this with the crystal structure of this heat with the enzyme. Now we are going for the heat to lead optimization, and some new derivatives of this compound has also been shipped from H3D, so they are being tested against DHODH, and we are going also to perform the cell-based antiviral assays with Tiago. We also develop many other machine learning and deep learning models, just to show a little bit this message passing feed-forward network. So it's a deep learning models that can learn with the data for phenotypic data for SARS-CoV-2. So we developed those models, and we were able to select other compounds for testing, such as a series from a diverse library from Flavio's lab. So we ended up with this compound with IC50 of 1.5 micromolar, and we also confirmed the activity or the heat with the crystal structure of the compound in the enzyme. So Flavio is performing the heat to lead optimization. We have many results for this as well, so I will just jump. And also this compound has been already tested with Tiago in Fiocruz, and it shows a very promising activity, inhibition of SARS-CoV-2 replication in Calu3 cells, as you can see here, compared to Breckina, which is a known DHODH inhibitor. So this is the COVID team. I'd like to thank all of them. Then we had some other collaborators that were joined later, such as Glaucius. And this is a very nice photo when last year we went there for the symposium. We had too much wine again there in Cape Town, very nice place. And now, just in my field, I don't know if I have 1 minute or so, but I will use them. Okay, Chris, I'd like just to introduce you, because all these projects, we are building these collaborative efforts. And then the COVID project fueled us, mainly Flavio, Christy, and myself, to start this new adventure, which is called CRAFT, Center for Research and Advancement of Fragments and Molecular Targets. So we are starting this, so our board, Flavio with the synthesis, Christy with the structural biology and screening of compounds, and myself with computer-assisted drug design and artificial intelligence. Our mission to educate a new generation of scientists to create a model for drug discovery in Brazil and to transform the science for society. We are now with this portfolio of disease, Chagas disease, Leishmaniasis, schisto, viral disease, and also we have our former project. They are also still ongoing on malaria and TB as well. And just to end, I'd like to show this 1 minute video. Let's see.
Sonho que transformou o propósito. Essa é a sensação ao ter fundado o CRAFT.
Criar novos fármacos não é fácil nem barato. E a longo prazo, nosso objetivo é contribuir para que o Brasil tenha cada vez mais opções de fármacos que falem português, que tem a nossa alcunha.
E é
com base nos valores da ética, da colaboração, do engajamento científico e da cidadania, nós criamos esse centro transformador da realidade brasileira.
O sonho que se concretizou é de todos nós e tem a nossa cara. É multicultural como o nosso Brasil, como a ciência deve ser.
And with that, I will finish. I'd like to thank everyone for your attention. Thank you.
Thank you, Carolina. That was a very nice presentation. So our next speaker is Dr. Aline Ramos Maia Loba. She is a researcher and coordinator of CRISPR-based target validation platform at the Instituto Butantan in Brazil. She got a bachelor in biomedical science at Federal University of Rio de Janeiro, and the PhD in biochemistry at University of São Paulo, with a period of a sandwich PhD internship at New Jersey Medical School in the United States. Postdoctoral at the Center of Excellence in New Target Discovery at the Institute Butantan, and currently she is the research and coordinator of the platform that she's going to be talking about. So Aline, please welcome.
Hello, everyone. Good morning. So I would like to thank you the invitation, the opportunity to be here and talking in the name of the whole group that has been working so hard in this project and other projects of the institution. But today I choose this project to talk because I wanted to tell you how we have been using Venos as a tool to find new targets, new promised targets for drug development. So before I start to talk about our center, our laboratory, and our research, I'd like to introduce the Butantan Institute because I realize that so much people here that is from another country, so I don't know if everyone know our institution, but the Butantan is a centenary institution in Brazil that we specialize in the venom and toxin, and nowadays we are one of the main institutions that public institutions that can produce a vaccine. And our laboratory is in the Department of Development and Innovation. This department is compounded for a lot of expertise laboratory that has a viral producer laboratory and a laboratory for developing vaccine, the laboratory that produce recombinant proteins and monoclonal. And our mission is just accelerating and improve technological process for the production and also to develop new products for that we can start to help in the health area. So now talking about Centergy. Centergy was started in 2016 and is a partnership between Butantan, GSK, and FAPESP. And the innovation of this project was the idea to use the venols and fractions venols and molecules derived from venols as a tool. Not the interesting of GSK is not the venol, it's just a tool to find a new target for drug discovery in immunoinflammatory disease. So to develop this project, we organize our center in 4 pillars that consist in the biobank and the screening platform. I'm just showing one of the parts of this screening, and the new target search omics, and for the last, the target validation. But in fact, we are organizing multiple platforms. So we have a specialized team for each platform, and we can, as I'm going to show you, we can pass all these platforms during the start of the projects and at the end. So starting with the biobank, we have a lot of venal that is from around the world. So all this venal is classified and characterized, and you will have a fingerprint based on the protein of these venals. And after this characterization, these venals pass for the screening step. And this is just a resume of our screening, because our interest is in osteoarthritis, and the interest of GSK is find a target, an anti-inflammatory target for using for develop a new drug for osteoarthritis. So the flow of the screening is it's basically we have some simple models and then the more complex models, and then we started the first validation and analysis. So we start with the crude venal and the fractionated venal, and we have some molecules, so we have some recombinant proteins and peptides derived from this recombinant protein that start with the inflammatory simple models, and then we pass for the complex model that is inflammasome and neurons, and then we started to analyze it, so just to validate the phenotype that we saw. And today I'm going just to show you with the result that we have using the peptides from two recombinant proteins that was identified in the venal of a caterpillar that is named Lonomia obliqua. And these two protein recombinant was interesting to analyze in the screen, because these proteins presented a cytoprotective activity in cell in stressed environment. So this recombinant protein was fragmented in several peptides. So you analyze this peptide in the screen that I'm going to show. So based on this, okay, that's I'm just showing this based on this picture that just to show the cells that compound the joint that we're going to study the osteoarthritis. We develop a screen based on this cell that represent the whole joint. So we have the macrophage that we saw huge infiltration of the macrophage during the process of the chronic disease. And we have the primary cell that is the chondrocyte, myoblast, and synovial site. And we incorporated the endothelial cells and lung cells during the pandemic, because we started to study SARS-CoV-2. And we established a model for neuro to study pain. So we established a neural model, in vitro cell model to study, to measure pain. So this is just a resume of the workflow that one of the screens that we established to study the anti-inflammatory activity of these venals and peptides and fractions. So that we have here, we established the model with inflammatory stimulus for each model, and stimulus of pain using collagen, decayed collagen in neurons. And then we analyze some release of some chemokine and chemokine, and with the balance of this chemokine, we decide which peptide presents some better response, anti-inflammatory response. And this is a resume of the results of one of the results using these peptides from the two proteins that we named Lozak and Lopapi. And from that result, we saw that this peptide presented anti-inflammatory, anti-nociceptive activity. And after that, we're going to the next step, is to confirm, to validate this peptide has this activity. So we start to use the omics, we use proteomics, and we can, using proteomics, we can just saw the enrichment of the signaling, so that we can, what we saw is that this peptide, but the figure is not, I couldn't put here all the results for all the molecular, for all the cell model that we have. That I put an example, that it is a macrophage, that we saw enrichment of anti-inflammatory signaling. And then these are important part of the project, because we have an agreement with GSK with the transfers of technology. So now that we have some peptide that could present anti-inflammatory activity, how we gonna to do, the targets that bind this peptide. So in this part, we, together with the GSK, they supervise, a specialized group from CellZone, supervised to establish a new technology, thank you, a new technology in our lab that is a chemoproteomic. So that's just, we modified the peptide, but we started to verify if the modified peptide has the same activity, and then we start to perform the chemoproteomic with this peptide. And this figure just to show the cross-link of the peptide with the proteins, and we analyze this peptide, these proteins that is able to bind to the peptides by using mass spectrometry. And with the result of the mass spectrometry, we have a short list of the proteins. And after that, this is the hard part that Professor Eldo explained, that is to analyzing the literature, because with the list, we have to decide which protein that binds the peptide could be trigger the anti-inflammatory response in the screening cell model. And this part, we always do this part together with GSK, and after this analysis, the huge analysis, we decide which target is going to be validated. So I'm not explaining, but here is just a flow that we are going to do, and he is just the step that we are now with several targets. We have now 3, 4, 5 targets running in several cell models. So with the first process of the validate, we make sure the specificity of this ligation. So that, here we have a gap, but we start to using the cross-link and the blot just to prove that this protein is the protein that is bind our peptide. And now we have to do the crystallography and some technique that prove the efficiency of the ligation of this peptide. And we start to, and after confirmation of this ligation of the protein, this interaction protein, the peptide, after we fishing, that used to do this expression, was we fishing the peptide, the protein that's possibly responsible for anti-inflammatory response, we start the functional assay to prove this gene is associated with the disease. So in this part, we have several techniques to use, but most of the time we use it to knock out, to use a CRISPR knockout for functional validation. And I'm going to just continue this part. And here, just to show that we use this platform, this organization of the platform, just to study other diseases. We start to study SARS-CoV during the pandemic. This is just one paper, and we have another one that is submission that we start to study the mechanism of action of the SARS-CoV, and we saw in this using several techniques with several expertise. We saw summarization that is important for the infection of the COVID. And this other paper is just to show how we work when we, in the Department of Development Innovation, how we work to solve a problem. So during the pandemic, all PI, all laboratories work together just to develop a serum that is able to neutralize COVID. So I would like to thank you, and I would like to thank in the name of the Director Innovation that is not here today, but she asked me to invite all of you to know the Butantan Institute and to visit us, and this is the email of her, and if anyone is interested, and just send an email for us.
Thanks, Aline. Great talk. So the last but not least speaker is my new friend, Maria Laura Bolognesi. She is professor of medicinal chemistry and director of the biotechnological, biocomputational, pharmaceutical, and pharmacological science PhD program at the University of Bologna. She's a passionate medicinal chemist with more than 25 years of experience in the neurodegenerative and neglected tropical disease fields. A common thread of her research is the development of chimeric small molecules resulting from the combination of two bioactive frameworks, including Proctox. She was awarded a Distinguished Visiting Professor Fellowship at the Complutense University of Madrid in 2009, a Visiting Professor Fellowship at University of Brasilia in 2014, and now in French, I'm getting Professor Invité at the Université Caen Normandie in 2018. See, I'm getting it. She's currently the president of the Medicinal Chemistry Division of the Italian Chemical Society, an associate editor of the Journal of Medicinal Chemistry, ACS, and serves in the advisory board of the European Federation of Medicinal Chemistry. So, please.
Okay, good morning, everyone. I'm truly pleased to be here, and I would like to thank Jardel and Luis, two new friends that I make with this trip, short but very intense. And yeah, I'm very happy, because today mine is not an easy task, because I was asked to talk about Proctox, because Luisa knew that I have this program ongoing because of Rocio, who is sitting there, was postdoc working at this project. So there was a spy here that knowing about this project. And so I said, okay, should I talk about a program like this one where we do not have final results? And I looked at the program, at the event, and then the hashtag is innovation together. So I said, yes, I should talk because this is the right audience, okay? So I expect comment from you on this very challenging project. So, yeah, I would like to focus the disease we are trying to address because the title was generic, but we are trying to develop Proctox for leishmaniasis. And this maybe puts leishmaniasis in a different perspective from what we heard so far, because leishmaniasis is also present in Italy. So now we should think about this disease as we heard the first day with a very too nice talk about climate change or whatever, that now this is not just a neglected tropical disease as this was as we intended this term some years ago. And then, yeah, there is a need of new drugs, and the idea that we are proposing is that we could use Protax to cure leishmaniasis. The general concept, as has been published here in this perspective, actually I should say that Rocio, I invited Rocio to write this perspective because we were waiting from the visa, we know the problem very well, and so we said, let's start to write in this perspective, so when you read it, so please consider that this was huge effort from Rocio before coming to Bologna. But anyway, yeah, Protax, what are Protax? You heard from the introduction that I'm very fond of chimeric molecules, and even Protax are bifunctional, also called chimeric molecules, because they have a part here in yellow, which is the POI, which stands for protein of interest ligand. Then there is a second part, E3 ligase binder. The two parts are connected via a linker. And the breakthrough when Protax were proposed more than 10 years ago, and was that they act by hijacking the proteasome, because they put together a protein of interest and the E3 ligase, and the protein of interest is ubiquinated and then degraded. So as you can understand, it's not like inhibiting a protein, but it's more like a knockdown of the protein. So this is quite innovative as a general idea with respect to the molecules that have been presented during these 2 days. But in spite of that, I was pushed to try to apply the idea in leishmaniasis, because there were already, when we started the project, molecules in clinical trials. And now there are 3 or 4, depends in the US or in China or whatever, but they have the potential to be truly therapeutic tools, drugs. And the project started in 2020, because there was a call in Italy on project high risk, high reward with no preliminary data. And so I said, okay, I want to propose my idea to tackle leishmaniasis with Protax. And this is what we did. So, but the problem is not, it's even more complex that you can guess. Why? First, oops, let's start. Yes, it's very tricky. But why, if I do something like that, maybe? Is it okay? Can you hear me? Yeah. Because I'm Italian, you know, I have to move myself, so. And my flight is at 6 p.m., so I cannot stand there because it's very risky. So, like any target-based approach, okay, this is something we know very well, we should start from a target. And then the first idea was, let's choose the target. But then we thought, the project is so risky, so we should start from a target which is well known. And so that's why we picked the trypanothione reductase, which is very well known, being involved in the metabolism of the leishmania, and it acts by scavenging the radicals. And because of that, it has a critical role in controlling the parasites redox homeostasis. And then it was very good, in our opinion, for our project, which we call the ProLeish, Protax for leishmaniasis. It was very good for ProLeish because it's validated, and also because this protein is absent in the humans. Actually, it has an homologue, but there is enough differences between the leishmania, trypanothione reductase, and the human homologue, which is the glutathione reductase. So we said this is a good starting point, and also this is a target that has been studied for a very long time. And so at the end, it also fits the needs of a PROTAC approach, because in spite it's a well-known target, then there are no very effective inhibitors. And this is because the binding site is quite large. And so it's very difficult to inhibit the enzyme and having a good parasitic effect. So in a certain sense, we can consider the trypanothione reductase as undruggable. So, and PROTACs fits very well the problems of undruggable targets, because they can be overcome because there is no inhibition, but just degradation. And so we started, and if you remember the structure of the molecules with the 3 different parts, then we tried to first to focus on the warheads, that is the POI ligand, then on the linker as in all the bivalent molecules is critical, in spite it has not a precise recognition role, but it's critical, we know that. But surely the most critical part was the one on the E3 ligase binders. So when we were writing the project, we were looking at the literature, and there was already enough information by saying that in the leishmania, there is this kind of ubiquitination system. So there were already E3 ligases that could be addressed and used for the PROTACs, okay? But then, okay, maybe there are the proteins, but we don't have the ligands, because these 2 guys here, thalidomide and VHL, are ligands that have been developed for the human E3 ligase. So what we did, we said, because we had to start, and so we needed a starting point. So what we decided to do is, okay, maybe even if we know that thalidomide is not the right ligand, because it can also recognize the human E3 ligase, we consider it a good starting point, a good entry point, because then we were not so much concerned about selectivity, because our idea was to get the selectivity because we were targeting a protein which is absent in the humans. So this was the idea. And even because there is plenty of literature that you can get selectivity even because of the formation of the ternary complex. So this is what we did. We docked we made the AlphaFold model of the protein, and then we went for used thalidomide. This was our first choice. And then we went also for the POI ligands. And because there were a lot of derivatives with these tricyclic scaffolds and even a lot of computational study on that, we decided to start from a structure like this one, because we could even identify the exit vector, because from there you can join the linker and then the E3 ligase binder. And Alessandra was the PhD student working on this project. She made the compounds, different linkers, and especially these are flexible. This one has less flexibility, and we tested the molecules. So the pipeline that we set up together with the Italian colleagues at the CNR, and we tested the activity against the protein. Actually, we tested the binding, because we wanted the binding were preserved, and so there was still the recognition. We also tested inhibitory activity, but this was a kind of surrogate test just to confirm. But then the problem was that when we went to the promastigote, because we were scared at the beginning about the selectivity. So we said, if we have the promastigote, we don't have the human proteome, and so let's start from that. But then no activity, and no activity, there were very flat results. And when even we moved to the mastigote models, it was the same. And so we said, wow, we were thinking that they were complex, but not so much. But then the idea was that, unfortunately, we picked up the wrong initial structure. Because, yes, we know that, but because protocs are such large molecules, the problem of permeation of membranes is so important, and no one has made protocs for parasites. So there were not even starting consideration to do. And what was even worse when we went to the literature, that, and this was done in combination even with studies by looking with the fluorescence, by exploiting the natural fluorescence of our molecule to see if they were internalized into the parasite, and they were. But the problem is that there were kind of aggregates inside the promastigotes, and we have understood that because these tricyclic scaffolds, when they have a peg connected, they can form nanostructure. And so these aggregates are so packed that they are internalized by endocytosis. This is what we guess. And then they stay there with no activity. So we were quite frustrated, but there are some lessons that we have learned. So we have started completely with the new POI ligands. And so I'm very in agreement with you, Carolina, because we have tried a fragment-based approach on that. And now we have these molecules. Here there is a fluorine, and we think to use this fluorine to link the linker and even the E3 ligase. So this is where we are. This is a perfect exit vector. So we want to continue in this direction, but especially because we are making a better model of the E3 ligase from the parasites. We want to even build a toolbox of ligands specific for the E3 ligase binder, and also the synthesis. Rocio is laughing because she knows how difficult is the synthesis, and thanks to the work of Rocio and many other students, now we have set up also the chemistry very well, because we are using a click chemistry, which is, of course, the best way to reduce the complexity of the chemistry. And yeah, just to conclude, what I want to say, yes, still protocs for leishmaniasis for me are still a dream, but if we want to make these dreams a reality, I believe it's critical to put efforts together. I trust a lot in open innovation, and you can see also from the picture of my group that it's in this rainbow mood. And there are two Brazilians, Rocio is here and Thiago is here. So I'm very positive that I can implement the project, maybe with the help of some of you. And thank you very much, obrigada.
Okay, so I'd like to invite the speakers to sit at the table. So, yeah, and we don't have a lot of time. Actually, I have an announcement. We are going to take another picture after this section. So we have about 10 minutes for the questions, and then we should be outside for another picture. We should start charging, I think, for so many pictures. And also, and we are gonna, after the picture, break for lunch, okay? So, questions? Rafael?
Hi, Maria. Nice talk. Thank you for teaching us about PROTAC. I would like to know, to hear your comments about how to identify the exit vector, and how do you plan to modify this vector in order to improve the biological activity?
You said that the vector, did I understand? Yeah, no, we have completely quit with that series. Now we are using completely different scaffolds, completely new, but I cannot disclose them because these are more promising, and so I don't know what I want to do with them. But my suggestion is that you should start with molecules that you know very well in terms of characterization of the binding post. So this is something that you can do very well with crystallography, whatever, but it's very crucial, in my opinion, to start with something, not, don't be offended, Carolina, but docking is not enough.
Definitely. I agree.
Oh, up there? Yeah.
Yes, hello. My question is for Professor Nakaya and Professor Horta. How do your companies synergize? How does your company or your institute in any way synergize with craft? Because I understand you both are in the data space, data exploration space. Do you envision any kind of synergy? And a second question would be how companies would use what you both do individually or in synergy to turn papers into invoices at the end of the day.
Well, actually, Craftsy is Carolina's initiative, and USP is a very big university with 6,000 professors. But I'm pretty sure I was going to discuss with her how can we do something together. I'm also work for a private hospital, so there we have an innovation branch which tries to translate the research into something. So we have partnerships at the private hospital with Pfizer and some other big techs that are running clinical trials and talking to researchers like me with basic science to make this happen. And I'm also a co-founder of a startup in Europe that is trying to use genomics and structural biology for pharmacogenomics. And that's another example. But I think Carolina can talk about the
No, it's just I am a big fan of Elder's work. I think almost everyone is. So yeah, definitely we should talk. I think we have synergy because he works more with the bioinformatics and network system biology, network medicine, precision medicine, and we can integrate this in the drug discovery pipeline. But as we are all discussing here since yesterday, is how we can build our, we can join our expertise and deliver a new drug, right? So this is the main point. I think we need to collaborate. We need to try to advance not only with your own expertise, but making huge efforts.
Up there,
Jair Siqueira Neto, University of California, San Diego. Really nice talks from all of you. I'm a big fan of AI, but I have perhaps a naive question. So when you're applying AI for predicting active compounds or predicting toxicity, it depends on the training set that you use. So your prediction will be as good as your training set. Now, taking that into account, what is the probability or likelihood for you to find a new mechanism of action if you're looking for an active compound against a parasite or a virus, or if there is a compound with a new mechanism of toxicity if that was not in your training data set? So what is your perspective on that? So it could be answered by Carolina or Elder. Thank you.
Well, this is a very good question, Jair. Thank you. Yeah, definitely. Artificial intelligence models, they are based on the data, right? But the good part is now that if we have data, good data, we need to have quality in this data. We need to treat this data. We need to see what we are going to give to the computer to build the models. If you give trash, you will receive trash. This is the major point, yeah? So the quality of the data, it's very important. We need to curate, to prepare this data. And regarding your question of the new mechanism of action, so if you are building models using phenotypic data, which we usually do, this is great because you are not target-based, so you are going to filter out compounds that will have activity in the parasite, in the virus, right? And then at the end, if you find active molecules experimentally validated, then you need to look for this mechanism of action, you know? So in my opinion, yes, we can discover new mechanisms if we are starting your models with phenotypic data.
Okay, one more question here. Please.
Hi, Mikhail from Sekimet again. I have a question for Professor Maria Laura. Protacs are quite a big thing right now, and we have a lot of information how the would bind to E3 ligases or VHL. Why would you avoid the human proteasome instead of using it to degrade the parasite? Like if you select, for example, a protein of interest that is outside or in the membrane, and then you just use all the information. Is it feasible, for example? Can you comment on that?
Yeah, thank you for the question. Maybe there are two questions into the same one. So regarding the idea of exploiting the human or the parasite system, I believe that there would be, our idea is to exploit the one from the parasite, but because it's highly conserved in all the eukaryotics, we need a very fine tuning of the binders for the E3 ligase, which at the moment, there is nothing. So we are starting to collect data, but because the binding site is very much conserved. So this is important. Regarding the two different localization of the proteins or whatever, I was even, this is something that intrigued me a lot, but then I even saw Protacs published on the amyloids, on the beta amyloids, where the amyloids plaques are outside. So, and even in good journals. So this is something, I don't know, yes, there is also a lot of junk, even regarding data, even of Protacs. So, but this is, to me, yes, it's more clear when the proteins and the system have the same localization. But then why there are papers on beta amyloids and even on the receptors, because maybe the receptors on the surface and then some way you can try to put them down the membrane. I don't know, but this is something which is intriguing me a lot. But I haven't seen yet a clear paper describing that. Thank you. Very, very, very nice question.
Okay, last question.
I have a question to Elder. Thank you for the presentation. It was really great. I saw that you have some work on getting connections from papers on the target and the mode of action of a compound. So how are you handling the indirect relationships between those nodes, and especially if it's a positive, negative, or neutral, or even not relevant, right? So how are you handling this the strength of the interaction? And a similar question would be, do you have any experience of working on generative AI for doing that, for generating new information based on the networks?
Well, great. Question for the dealing with papers, the machine learning can detect the expression, right? So if it prohibits expression or inhibiting, it's negative. So you can take this into account. So we did one with IBM Watson that's already give the directions, and the other one we just use we do not use this information. So we count the papers. But regarding the question, the previous question, a lot of papers were actually garbage, right? So we use actually this approach as a screening process. But at the end, if we're going to publish, we check by hands each one of these connections to see if this is true. before we publish, but at least you reduce the amount of stuff to look at. For generative AI, well, we are using this for the computer vision part of the lab, for example, to generate chest x-rays, because we are trying to make a CT scan from x-rays, and then we have to train on images that actually do not exist, so we need more image. So also the previous question regarding the big data, actually we're now focused to more smart data, which is like, it's better to have smaller data with good quality, like well controlled and a lot of information than trying just to put a lot of not useful information.
Okay, thank you. I think we ran out of time, so let's thank the speakers. Okay, and let's take the photo outside.
Yes, here.
Good afternoon, everyone. We'll now start the afternoon section. And this section will go from 2 to 3:30, and then we'll have a break. Each speaker will have 20 minutes to talk. And today we'll have 4 speakers from the pharmaceutical industry representing their global health sectors. One of the speakers, the national one from Europharma, Karini, was not able to make it because of a family emergency. So we'll keep the 4 talks and then we'll open for questions. And the speakers from today, they are my heroes for advancing compounds to clinical development. I think being in academia, this is our dream when we work on basic science, right? So I work here at Unicamp at the Center for Medicinal Chemistry, and we know how the interactions with the pharmaceutical company are essential to drive our research to the patients. And also what we have already discussed yesterday is how important the partnerships are between university and the private sector. If we think how the global pharmaceutical companies have made significant contributions to the fight against neglected tropical diseases, we can think of many ways how this happens. For example, by drug donations. Here we have Dr. Fabian Guzowski, who was involved in the distribution at zero cost of diethyl carbamazine, a drug used for treatment of filariasis, for example. We also see how the pharmaceutical companies involved in access programs for open science or free drugs and accessible drugs. We know they also work on advocacy and fundraising from government and private agencies to allow this accessibility. And they develop their internal R&D programs and invest in capacity building. I think the role of the pharmaceutical companies is very clear to us for this kind of public, right, in organizing the public-private partnerships as we have been talking about. And by having their internal programs and interacting with all the other sectors. And I will introduce the first speaker, Dr. Fabian Guzowski. He's Vice President of Global Health Research at Eisai company in Japan. He manages a team responsible for drug discovery and development portfolio with access in malaria, tuberculosis, and neglected tropical diseases. And he also has many collaborations with academia around the world and is a member of the executive group of Global Alliance for Elimination of Lymphatic Filariasis, and the Executive Council supporting the Gates Foundation. With that, I ask you to welcome Dr. Fabio.
Thank you very much for the introduction. Thank you for the invitation and congratulations on the occasion of the anniversary of this very impressive initiative. Also, thank you for the flexibility of moving me on top of the program. I need to catch a plane later, so. I work for Eisai. Eisai is a Japanese pharmaceutical company. We are, the company is headquartered in Japan. I do not work in Japan, I work in the United States. Part of our company philosophy is what we call the HHC mission, that stands for Human Health Care. And we define this in many ways, but in what is focused towards global health, we have two components. We call this social good and efficiency. Social good is to develop and deliver new medicines for underserved patient populations. And by efficiency is the most practical and sustainable and realistic way of doing this. We leverage external funding. We have established collaborations with leading academic groups around the world, as well as nonprofits and pharma partners. And finally, we apply innovative technology approaches. So that hopefully will result in a sustainable way that generates high impact and global health leadership. Using this model, we have been working for the last 11 to 12 years to build a very large pipeline of projects. Some of those projects were initiated at a very early stage, and I'm glad to say that several of them have reached a clinical development stage, and I'm going to share with you a couple of examples of these projects, as well as a couple of programs that we have initiated more recently, where we are attempting to utilize novel technologies, typically not used in the area of global health for the high cost, but I think we may have a way to undertake this challenge. The first one is a compound called E1224. I will give you a little bit of history of this compound. The molecule is the phosphoracetamol, was developed at Eisai a long time ago as an antifungal. It has a history of out-licensed and licensed back to Eisai for a number of reasons. There were phase 1 studies performed with that, but ultimately was found out that there were physicochemical problems with the original molecule. A prodrug was developed that is called phosphoracetamol. Phosphoracetamol, in the presence of alkaline phosphatase, releases the parent drug, and that results in a much more soluble compound that is bioavailable and pharmacokinetically appropriate. In collaboration with DNDi, we have initiated 2 clinical programs utilizing this molecule, one in Chagas disease and the second one in mycetoma. And today I will give you a little bit more information about the mycetoma study, which is ongoing. Mycetoma is a fungal, in one of its forms, is a fungal disease that is occasionally known as the most neglected of the neglected diseases. It was only recognized by the WHO as a member of the NTD list in 2016. Its geographical location is between the tropics, in Africa, Latin America, and Southeast Asia. The advanced form of the disease is quite devastating, resulting in amputations. There is no clinical trials to the day with antifungal agents that have shown efficacy. And a few years ago, Professor Fahal, who runs the Mycetoma Research Center in Khartoum in Sudan, and his collaborators demonstrated that in vitro systems, fosforoquinonazole showed the most potent activity against the fungal agent causing mycetoma by several orders of magnitude. With this information in hand, and given that we already had a lot of safety information, even phase 1 studies in humans, in collaboration with DNDi, we initiated clinical development. I should say that DNDi took control of the clinical development. DNDi is the IND holder, whereas ASI supplied the clinical trial material and provided other types of in-kind contributions. So the study is still ongoing. We have not released the results. There are several positive indications, but this is one of the examples where we can repurpose an existing drug to a new indication with somehow optimistic outcomes. The next molecule I want to cover is called E1018. This is a result of a collaboration between ASI and the Broad Institute, Professor Schreiber's lab. In addition, there are members at the SCRIP Research Institute, and more recently, for the clinical developments, we have the opportunity to work with members of the Walter Reed Institute and the Cambridge, which is a Kenyan Medical Research Institute, in a site located in Kisumu in Kenya. This molecule is a first-in-class inhibitor of phenylalanine aronase synthase as an antimalarial. It's the first ASI-sponsored IND program in global health. It's our first collaboration with this Kenya group, and is the first member of the first example of the collaboration that we had with the Broad, with Schreiber's lab, to enter the clinic. So it's breaking ground in a number of things. So a little bit more about the molecule. This is the structure. The compound is nanomolar potent in vitro against malaria. The all in vitro properties have been optimized to very, very good levels. And what is most interesting about the compound is that in an animal model of malaria, a single dose treatment with this compound can result in a cure. And in the space of malaria, this is the type of profile that we are looking for, and therefore we are quite excited about the prospects of this agent. As I said, this is an inhibitor of phenylalanine aronase synthase. This is the catalytic, the reaction of coupling phenylalanine with the tRNA. This is the enzyme, and the compound was discovered to be an inhibitor of this biochemical step. In 2021, this was corroborated, and it turns out that a compound of the series is very clearly crystallized in the catalytic pocket of the enzyme. So not only this provides reassurance that activity on the target, but allowed us to initiate a second generation synthetic programs to improve over the existing compound. Jumping ahead a little bit, we completed the preclinical package. We're in the process of IND submission, and we have partnered, as I said, with the Walter Reed Institute and the Combewa Clinical Research Center and Cambridge in Kisumu. We visited the site at the edge of Lake Victoria. This is also quite a novelty. Typically, for global health products, companies tend to do phase 1 studies with healthy volunteers in Western countries. We were suggested many times that why should we do that? Why, if we are going to develop a compound for an endemic region, why don't we try to run phase 1 studies there? Well, this is what we did, and we had the fortune to establish a very good collaboration with this team, and this is what we are hoping to deliver. I'm going to switch a little bit to another topic. I will like to cover a couple of areas that we are started recently trying to utilize innovative technology approaches in global health products. The first program that I would like to share with you is an AI-enhanced screening for Chagas disease that we have started about a year ago in collaboration with 2 groups in Argentina, at the Universidad Nacional de La Plata with Professor Alan Talevi, and at INGEVI in Buenos Aires with Guillermo Alonso. The purpose of this is to address the need in Chagas, which in short we need an effective way to deal with the chronic form of the disease for which there is no adequate drug. And in addition, as we know, the drugs that are being used are very old and not lacking some undesirable properties. There's urgent need for new drugs, and what the team is doing is generating an AI system to facilitate resource-efficient screening. For that purpose, we are utilizing machine learning models to reduce the cost and time of screening. We apply target-based machine learning approach to a particular target family, which is the phosphodiesterase, and the focus is first to try to repurpose existing drugs that perhaps we did not have the ability to predict that would be effective in Chagas. And second, as the second option, to utilize ASI library as a source for new hits. As I said, the partners are Talevi and Alonso, and their role is to generate the machine learning model, to perform the experimental assays, and also, given that Chagas is endemic in Argentina, they provide the endemic country perspective. At ASI, we generate an orthologous machine learning model and provide the pharmaceutical capabilities. This is where we are in this project. We completed the first stage of target validation. This was a very, very interesting piece of the study in which the team in heavy utilizing CRISPR-Cas in trypanosoma were able to show the essentiality of the phosphodiesterase members of the family. And we're in the middle actually of generating the machine learning models. And there's two types of model. There are ligand-based models and CPI models run at ASI. So what we have the capacity of doing now with two different types of models to define a pocket through molecular docking, and utilizing the properties of both models, do a hit selection from both repurposing libraries and ASI libraries to increase the chances of generating hits. In the near future, once this is completed, we will engage in experimental profiling and ultimately in vivo demonstration of activity in Chagas models. The last project I would like to share with you is a new approach to deal with prevention of malaria, utilizing antisense oligonucleotides for malaria prophylaxis. So there is a need for effective, long-lasting protection against malaria. The burden is quite high. There are 600,000 deaths yearly, and largely children under the age of 5. There are some solutions ongoing, but they are not completely satisfactory. Mass drug administration that has been proposed seems logistically challenging. Vaccines are available, but they have modest efficacy and repeat dosing. So we are looking for a long-acting antisense oligo that can migrate to the liver upon administration, given that the liver is the first organ where the malaria parasite accumulates and propagates. The utilization of ISOs provides the ability to design molecules against a variety of targets with high selectivity. And we have, in addition, a quite interesting platform at ASI, whereas we believe that adding stereo control to oligonucleotides could result in a quantum leap in terms of the activity of against the target. Our partner is Professor Elizabeth Winsler at UCSD. They are experts in malaria target. They have all the screening tools, and ASI, of course, providing the chemistry background, knowledge, and compounds. I would like to finish there. I would like to acknowledge our collaborators, again from in the Chagas program, from La Plata and INGEVI, Elizabeth Winsler in San Diego, Professor Cajal and his team in Sudan and in the Netherlands, and of course the DNDi team without whom this would not be impossible. In the 1018 program, we partner with Walter Reed, the Broad, and us have been working on this for several years now, and of course, the Kenya site and Scripps. And finally, the supporting organizations, the GHIT, or the Global Health Innovative Technologies Fund, is a Japanese organization that funds proposals directed specifically to global health. And we have been successful in securing funding for several of the projects that I've shown in the pipeline. The CDMRP is the division of the US Department of Defense that provides grants, and we were able to secure funding for the malaria project. In addition, we have received support from the Gates Foundation and the Medical Research Council in the UK and the NIH in the US. Thank you very much.
Do you have to leave right now or? Okay, because otherwise we would open for a question. So the challenge here. Our next speaker is Michael Schrimpf. He's research fellow at AbbVie in the United States with 25 years of experience as medicinal chemistry and project director at Abbott and AbbVie. His PhD is in organic chemistry from the University of Wisconsin-Madison. His current team works on antiviral drug discovery projects and directed neglected disease program. He has been awarded more than 30 US patents and has also helped in advance 15 compounds to the clinical development. He has been recognized 4 times with the AbbVie President's Award, and he can tell us a little bit more today about his trajectory.
All right, thank you so much. I just want to say that it's an honor to be here today to join in this celebration. I want to congratulate the LOLA team and DNDi and all the partners for what you've accomplished over these 10 years. Your passion and your vision is really inspiring, and I can't wait to see what you do in the years to come. So I'm here today to tell you about AbbVie's program in river blindness. Before I do that, just briefly describe our approach to our work in neglected tropical diseases. So like some of the other pharma that have been up here, we are signatories to the Kigali Declaration. We have support from our CEO all the way down to the company to do the work we do. We talked a lot about collaboration, and we see things the same way. We don't invest in the disease biology. We leave that to the experts, like many of you. And what we do is the thing that we do best, which is the drug discovery and development. I'll also say that in contrast to some other organizations, most of our work is done by people who are not assigned this as their job responsibility. They volunteer for it. It's something they do on top of their normal responsibilities. And I actually love that aspect of it. The people that do this are very committed, and they really believe in what we're doing. This shows some of the areas that we're in. On the preclinical side, mostly tuberculosis. Some work here in support of LOLA on leash and Chagas disease. And I'll be talking about onchocerciasis today. So I imagine many of you are familiar with river blindness. This is a really debilitating disease caused by infection with a parasitic worm, Onchocerca volvulus. Humans are the host for this worm. It's transmitted from human to human via a black fly. The black flies live near rapidly moving water, hence the name river blindness. So the infection cycle, the fly, I'll say bites one person who's infected. Some of the worm goes into the fly. Several larval stages happen in the fly, and then it's transmitted to a second person. I can't see my slides very well. Okay, so the main effects of this are in the skin and in the eyes. So the worms, the adult worms can live in these nodules. Let's see. Like this. There are also some other skin inflammation and irritation. And then the terrible part, when the microfilaria get into the eyes, if they get into the eyes and they die, the body's response to that death of the worms is what actually results in the blindness. So it's estimated as of 2017 that about 21 million people are infected, and over 1 million have visual impairment or blindness. And you can see many millions more are at risk. The disease is most prevalent in sub-Saharan Africa, with some pockets near the Brazil-Venezuela border. This map is a bit outdated. There were other infection sites in Mexico, Guatemala, Colombia, Ecuador. Through some aggressive campaigns there, those areas are now free of river blindness, which is very encouraging. So this disease used to be considered by the WHO as a public health problem, and so the focus was mainly on the areas of highest concentration. That changed a few years ago. Now the goal is total elimination of transmission. So all areas, even those with smaller infection rates, are being targeted. So there is a treatment, a moderately effective treatment, and that is mass drug administration with ivermectin. It takes 6 to 12 months, or it has to be given every 6 to 12 months, because ivermectin only temporarily sterilizes the worms and kills the immature microfilaria. It does not kill the adults. And the adults can live for many years, say 7 to 15 years. So you have to keep giving the drug repeatedly. And that has challenges. So, there's poor compliance because there are side effects of this. It's just logistical problems with giving this drug to large numbers of people every 6 months to a year. Also importantly, ivermectin is contraindicated in areas that are also have common infections with LOLA, a second worm. So what happens there is ivermectin kills the LOLA, and it's such a rapid death of the worms that the body's response there can actually kill the person who had the LOLA co-infection. So there's now a strategy for that, to test people before they're given ivermectin to make sure their LOLA levels are low. But that still is a problem with this mass drug administration. So as a high priority, it has been identified that we would like a better drug that will kill the adult worms and really end the cycle. So it turns out that the worms have a symbiotic bacterium, Wolbachia. And so this has led to a really, I think, clever strategy. The worms need the Wolbachia in order for embryogenesis and survival. So the strategy is, if you are able to kill the bacteria, you should be able to kill the worms. Doxycycline has been shown to be effective in this way. So it is an antibiotic that kills the Wolbachia. However, that takes, That takes 4 months, 4 weeks of treatment, at least 4 weeks of treatment. And doxycycline is contraindicated in children and pregnant women, which is a very large segment of the population. So the target profile for this program was to find a new anti-Wolbachia agent that, first of all, will work in 7 days or less, 7 to 10 days, and better safety than doxycycline. So it can be given to all people who have the disease. And this was a collaboration with AWOL and DNDi. So AbbVie's involvement began about 10 years ago when we had many old antibacterial programs, and we had compounds in our collection. And a small set of those, 129, were sent to the Liverpool School of Tropical Medicine for a Wolbachia assay. One compound really stood out. That's tylosin A. And I actually dug out the original data set here. That's the gray compound. This is the dose response. And red is doxycycline. So this compound really stood out as being a potent anti-Wolbachia agent. You can see the structure of it here. This tylosin A. And very interestingly, this is an antibiotic that is used in animals, in veterinary medicine, in pets. In fact, you can buy this in the US, you can go to your local pet store and buy this. So it's readily available and very inexpensive, which is really important when you're talking about developing a drug for this population. So in in an in vivo assay, this compound tylosin A was shown to be as effective as doxycycline. So on the y-axis of this plot here, this is worm length. This is the control in blue. This is doxycycline in red. And then this is tylosin A, 7 days given IP. The white bar is the tylosin given orally. You can see it's much less effective given orally. And the reason for that, when we looked at the PK of this compound, is that it's not orally bioavailable in mouse. So I had to smile this morning when we were talking about natural product drug development, because here's a great example of where that can be done. So if you look at the structure, obviously there are lots of hydroxyls, polar hydroxyls around this molecule. And the hypothesis was that this compound is poorly absorbed because it has poor permeability. And the MedChem strategy was to try to mask some of those hydroxyls, see if we can improve the permeability. That's where this person here, Tom von Geldern, came into the picture, and I think Tom is known to some of you. Yes. So Tom is an extremely talented medicinal chemist, and he took on this challenge. He started derivatizing the different hydroxyls of this molecule to find the ones where substitution there would be tolerated, and to see if any of those improved permeability. He made about 150 compounds, and I can't go through the whole story, I wish I could, but basically at the end he found that this particular hydroxyl, the 4 double prime hydroxyl, could be substituted, and the best compound he made was this parafluorobenzyl ether. And that's a compound that became ABV4083 or flubentylosin, also sometimes called Tylomat. I should point out that I mentioned this program is done mostly by volunteers. And Tom, really kind of an exceptional example, had already retired from AbbVie. And he came in a couple days a week on his own time and did this just as a volunteer. Really remarkable story. So once we had the candidate identified, we had to figure out how to make large amounts of it for development. And that's where a second AbbVie retiree came in. That's Howard Morton. He had been a process chemist, had retired. He did the same thing. He came in a couple days a week on his own time and developed a really nice 3-step synthesis of this molecule, starting with the really inexpensive tylosin A. So it's a very potent compound. So not only did the benzyl ether improve the permeability, it also dramatically improved the potency. So it's much more active than tylosin A itself, much more potent than doxycycline. And this is also summarizing its activity across a number of different models. So the problem is mice are not susceptible to infection with the onchocerciasis worms. So you have to go through either using Wolbachia infection in different worms, or you have to look in different animal species. So there's really not an ideal animal model for river blindness, but if we look across all these different models, we saw that ABV4083 has really good activity in all of them. So this was the phase 1 plan, kind of a standard dose escalating plan, single dose. When we got to 1,000, there was a signal, a safety signal, ALT and AST elevations. And after some work, we determined that sticking at 400 mgs was tolerated, was safe and tolerated. So that was the dose that was chosen to go forward. It's also, based on the modeling, expected to be above the unbound levels will be above what we expect that we needed for efficacy. So this is the clinical trial design. So there were a number of arms, different dosing, lengths of dosing, combinations with albendazole, which is another drug that can work in river blindness. So we're looking at single agent, looking at the combination, looking at lengths of dosing. This is the study population. So this was the phase 2 study here was done in the Democratic Republic of the Congo, supported by DNDi, ran the studies. The patients needed to have at least one nodule, because that's where we were going to be sampling for efficacy. And they could not have been on other treatments in the recent past. And as a sort of success criteria, we were targeting 77% depletion of Wolbachia in surviving worms at 6 months. Pharmacokinetics were exactly what we expected based on our phase 1 study. We saw levels that we thought were needed for efficacy. Interestingly, so there was a kind of unique PK method that was developed for this. So the samples are spotted on paper, and the paper could be shipped back to the United States, to Chicago for analysis. So they didn't have to do any sample processing at the site. So here are the results, and these are fairly new results. We got these in June, I believe. So you're shown 7 days of 4083. These are the numbers of worms. Well, here's the total number of worms in that sample group. Here's the number that had no Wolbachia, and this is the percentage of worms with no Wolbachia. So we were targeting 77% reduction in Wolbachia. And to our great disappointment, none of the dosing groups showed any efficacy. When I agreed to give this presentation, I'd hoped I'd be delivering a much different message, and really expected I would be delivering a very different message. But this, I think, was a pretty clear-cut answer that the compound did not work. Just for comparison, here are some data from a different, unrelated study of doxycycline in onchocerciasis. And you can see here, the top line, and maybe you can't see, it might be too small, is doxycycline 4 weeks, which is the standard treatment. And these columns here are many, let's see, this is the number of living adult worms that have none, or no, sort of have many Wolbachia, have few Wolbachia, or have no Wolbachia. So you can see, in these dosing groups here, especially the standard one, there were no worms with many, only one with few, and most of them, 70 of those had none. So it's for comparison, you can see as a positive control. So that's very disappointing. As I said, not the message I was hoping to deliver. And we're all scratching our heads, what happened? And I don't think we have an answer yet. We know that the plasma levels are what we expected would be efficacious. We kind of wondered, is the exposure in the nodules or in the worms insufficient? That's a possibility. Maybe we didn't dose long enough. Maybe the Wolbachia were not knocked down far enough, and they were able to recover, because you notice 6 months. We didn't check until 6 months. Maybe there was some difference in the Wolbachia that were in the study versus what we had used in the lab. I guess we don't know. But people are looking into this, because it's something that could inform future efforts in this area. So, I think we all know this. Drug development is a risky business with high attrition rates. Fortunately, DNDi and I think other people may have other trials going forward with different agents. I mentioned the issue of the animal models. So compound worked well in all the animal models we tried. The animal models were just not predictive in this case. So in spite of the disappointing news, we were really encouraged by the partnership we had with DNDi and with other partners. This is sort of an example we'd like to follow for future collaborations in the NTD area. So I'll conclude there. I want to thank, there's too many people to list, so I just say the hundreds of AbbVie employees and other collaborators who worked on this project. And I highlight here Dale Kempf, so I know many of you know Dale as well. He started the NTD program at AbbVie, and he was really the guiding force behind this whole project for its entire history. So I acknowledge him, and I want to thank you very much for your attention.
Hello. Our next speaker is Dr. Srinivasa. Rao, Director of Global Health of Novartis Biomedical Research in Emeryville, California, USA. He's leading multiple drug discovery and developmental projects against various neglected diseases, which includes parasitic, bacterial, and viral diseases. I saw in his CV that it actually came as a drug hunter. I imagine you call yourself a drug hunter, right? And also a basic molecular biologist defining the best targets for each case. I think that combination of knowing enough of the biology, right, to look at different pathways and have this knowledge of seeing what's a good target and how you match that with the best drugs is an art, right? And it seems that it's a lot of fun for you, right? And an example of one of the molecules mentioned recently is LXE408, which is entering clinical trial against leishmaniasis with the potential to be assessed against Chagas disease too.
Hello? Yeah. Thank you very much. And I would like to take this opportunity to thank DNDi and LOLA Consortium for inviting me. Jardel and Luisa, thanks for inviting me. Also, I just wanted to, take you through one of the stories what we have developed over the last decade today, but we have multiple other programs which are running simultaneously. So, at Global Health in Novartis, we are not only working on leishmania and Chagas disease, we are also working on malaria, and we have seen a couple of success stories in malaria, and we are running a phase 3 trial with CAF-LUM with the help of MMV and Wellcome Trust. Hopefully, we'll see some registration candidates there. We are also working on cryptosporidiasis. We have a candidate which is ready to be tested in man. We have completed phase 1. Hopefully that will reach the patient soon. Recently, we also started working on pandemic and epidemic diseases of interest. And here we are mainly focusing on 3 viral families is Flaviviridae, which has dengue. And then we have COVID coronavirus family, as well as henipaviruses. Henipavirus is a very deadly disease. If you get it, 70% chance of death. So these are the viruses we're working on. And I just want to mention that we do have a candidate for dengue, which is entering phase 2 trials as we speak. So looking forward to some of these candidates reaching and having impact on patients. One of the concept which we had discussion yesterday, I always call ourselves collaborative approach in global health, but there is no competition here. We all need to work together in order to deliver clinical candidates. What I wanted to highlight, we are not only collaborating externally, but we are also collaborating heavily internally. So in drug discovery, you have 3 different groups. One is a discovery group where you identify a target, develop a molecule, test its efficacy, test its toxicology, and bring it all the way to phase 1 and phase 2, a proof of concept. Then there is a second group which takes over, which is a development group. They run phase 2B, phase 3 trials in order to register the compound. And there's a last group which is very critical, which is global access, right? So when you make a drug, it has to reach all the patients. So we have all the 3 divisions within Novartis. When we initiate a project, right from then, we have this end-to-end approach. They know we are working on this disease. So in those endemic countries, they start really planning in case if we are successful, how do we reach the patients, every patient as soon as we launch the drug. And I just want to really highlight a lot of collaborators. I just could put only a few of them here. We have extensive funding from Wellcome Trust, NIH for Pandemic Preparedness Grant, MMV for malaria, and Bill and Melinda Gates Foundation for Malaria, Malda Project, part of the Malda Consortium. And within this region, we have started heavily collaborating with some of the people in the audience. Professor Anna is here, Carolina is here, Jardel, and few more people We are heavily collaborating in order to deliver candidates for Chagas. I do not want to go through the canidoplastic diseases, all of you know very well, but I just want to highlight two important points. What is that is these three diseases are really neglected diseases, and more than 1 billion people living across 80 different nations, crossing 5 continents are at risk of getting these diseases, and there is around 30 to 40,000 people death, but there is a majority of the population, millions of people are living in morbidity, which needs to be alleviated. So the current existing drugs, this once again, I'm teaching to the choir, everybody knows, existing drugs are mainly repurposed drugs. They were not developed as a direct antiparasitic for Leishmania or Chagas. So there is, and these drugs are decades old. So there is an urgent need for developing safe, short course oral therapies, which are direct antiparasitics, which are specifically tailor-made for Leishmania and Chagas. So our approach was, our approach for finding novel drugs or direct antiparasitics was phenotypic based approach. So the initial challenge, what we thought was, as you heard in the yesterday and a couple of talks, developing a candidate from the time of identifying a molecule to take it to the registration, it costs you $1 billion+. So we thought, how about looking for a common mechanism of action which is conserved across these 3 parasites, and we could reduce the development cost for those molecules. So keeping that in mind, as you know, all these 3 parasites belong to canidoplastic group. The whole genome sequencing suggests anywhere 60 to 80% homology. If we can hit a target which is conserved across these 3 parasites, we may be able to develop a single molecule as long as the molecule enters where these parasites are hiding in the tissues and clear them off. So that was a kind of a hypothesis. So we went ahead with 2 million compound library screen in the beginning against Trypanosoma brucei, Leishmania donomani, and Trypanosoma cruzi. We identified multiple compounds, but our focus was on pan-canidoplastic hits, which is around 85 to 100 compounds. The other approaches what we took was, in order to expedite the drug discovery process, we kept our flowchart very lean. We focused right from the beginning compounds which are cidal, and there is also an assay which is called as washout assay, where you incubate the compounds with parasites and wash out the compounds, and they should not relapse back. So these were important criteria which we adopted right at the beginning. The other criteria which we adopted was, as soon as we identified a certain compound list, we did a structure activity relationship based on Novartis archive, and we took a couple of compounds very quickly into mouse efficacy studies, evaluated the potential of these molecules to clear parasites in an animal model, measured the PK at different concentrations, and see whether there is a PK/PD correlation. And that helped us to prioritize the series to go for a real lead optimization. On top of that, we rapidly adopted lead optimization toxicology studies in order to find out whether the series has any other safety liability, going directly into rat at the beginning so that we know what are the liabilities, so medicine and chemistry can start fixing. When we were doing this, the most important point, since I'm a biologist, was target identification. We spent a lot of time internally as well as with academic partners, quickly identify the target so we can develop target-based assays, not only biochemical assays, but cell-based target assays to make sure that the compounds what medicinal chemists are making is hitting the right target and bringing about the death of parasites. And in fact, we did start multiple target-based programs in order to find novel chemical entities using this approach. So I'm not doing a justice by just putting 3 compounds here. We made 3,000 molecules in this series. Our medicinal chemists were really ramping up, cranking molecules like this, and we were generating data on a weekly basis, providing them clear-cut guidance on their activity on parasites as well as their physicochemical properties. From the first molecule to second molecule, we had already made around 1,000 molecules, and the second molecule was showing activity in mouse models, having good PK properties, but solubility was not good. So we made the last molecule, which is LXC408, where it was meeting with promising activity across parasites, non-cytotoxic, and also showing better solubility and PK properties. So as I was mentioning, we focused on addressing what is the target for this particular series of compounds. These belong to triazolopyrimidine series of compounds. We used a standard approach of raising resistant mutants in Trypanosoma brucei and Trypanosoma cruzi. And it took nearly 6 months to 2 years, depending on the strain. And after that, we did whole genome sequencing and tried to locate where the mutation is. For our good feature, we did not only create with one molecule of triazolopyrimidine, we did with multiple different molecules, and we looked for a common mutation so that the target is more conserved. So proteasome beta-4 pocket was having a mutation, beta-4 F24L, and beta-4 lies very close to beta-5, which is a chymotrypsin activity of 20S proteasome. So we went ahead, showed biochemically that LXC408 and its molecules are indeed inhibiting proteasome, that is chymotrypsin-like activity. And we made a mutation in beta-4, that is F24L pocket, suggesting that there is a shift in the IC50. So indeed, these molecules are binding into that pocket. Finally, we carried out cryo-electron microscopy, and where we noticed triazolopyrimidine is indeed binding in between beta-4 and beta-5 pocket. The molecule which is on the top, it is bortezomib, is a known proteasome inhibitor. It binds differently compared to LXC408, suggesting bortezomib is a known proteasome inhibitor of humans, and ours is not inhibiting human. So this is a series of assays we did on multiple different parasites: Leishmania donovani, Leishmania infantum, Leishmania major, Leishmania brasiliensis, and few more. And this was done in collaboration with multiple academic groups. I have not put all the list. DNDi helped significantly in generating this data. It was active across different species, including T. brucei and T. cruzi. And we also ran 20S proteasome activity across these three parasites. As you can see, they are active on T. brucei, T. cruzi, and Leishmania, but they are having a significantly higher activity, more than 20 micromolar on humans, suggesting that they are pan-kinetoplastid specific proteasome inhibitors. So once we had the molecule, we ran multiple different efficacy models. Here I'm showing visceral leishmaniasis model run in Balsi mice. So as you can see, 1 milligram was sufficient enough to achieve 95% reduction in parasitemia measured by qPCR in liver. And we also did 3 milligrams and 10 milligrams, they were reaching at the bottom. And we collected PK samples. As from the PK samples, you can see that 70% coverage of EC50, pre-EC50 was sufficient enough to achieve around 95% reduction, which is similar to the standard of care miltefosine. So here are a few more models. We ran LXC408 against cutaneous leishmaniasis model. We ran both old world as well as new world model, and 3 milligram was sufficient enough to achieve complete suppression of parasitemia in the suppression model of L major, and 20 milligram was sufficient enough to see all the 6 mice relapse-free lesion there. And although in this model we did see 3 out of 6 mice coming for ambiosome treated group. Finally, we used L. guyanensis. This model was run WRAIR, Walter Reed Institute, and we saw 20 milligrams is able to clearly reduce that lesion. So since this molecule had already entered phase 1, we couldn't run a bioluminescence model for Chagas, but we ran a previous model which was developed by our San Diego colleagues, where we are measuring parasitemia in blood by qPCR, and also we collect heart and colon region after the end of the treatment of immunosuppression. As you can see, 3, 10, and 30 milligrams were able to achieve below limit of detection of parasitemia. One mice relapsed, it was slightly higher in 3 milligrams before immunosuppression. But after 4 rounds of immunosuppression, what we did in immunosuppression is every week, 2 rounds of 200 milligram IP dose of cyclophosphamide was given. So totally 8 doses of cyclophosphamide, suggesting that we really immunosuppressed all the mice. In that model, as you can see, 30 milligram achieved 7 out of 8 mice cure. And there was absolutely no parasite in colon and heart. So once we have a molecule which is showing potency, in vitro potency and efficacy in the mouse models, believing that we can translate it to human, so we went ahead, did PK studies in multiple different species, mouse, rat, monkeys, and dogs. As you can see, The clearance is low to moderate, and we have half-life ranging anywhere between 3 to 10 hours, and oral bioavailability is ranging anywhere between 30 to 70%. This was sufficient enough for us to move to the next stage. The next stage in drug discovery is, before we take it to patients, we need to do toxicology studies, and we did run toxicology studies for this in both non-GLP and GLP. GLP 4-week toxicology was done in rat and monkeys. LXC behaved well. We had a sufficient TI of 10 with respect to visceral leishmaniasis. So that's why we took first for VL. And then we had to run healthy volunteer studies, as you saw from previous talk. We did all the way. And then currently it is in adults, and after we are successful in adults, then we go to children. So this is the plan for phase 1, what we adopted, a single ascending dose, anywhere ranging from 10 milligrams all the way to 600 milligrams we went. And for the MED multiple ascending dose, we gave 10 days of duration of treatment, all the way from 10 milligrams all the way to 600 milligrams. Here is a summary of preliminary pharmacokinetics as well as safety. As you can see, LXC was rapidly absorbed. Tmax was anywhere between 3 to 4 hours across different doses, and half-life was anywhere between 18 to 20 hours. So one day daily dosing is sufficient enough to reach half-lives. And plasma concentration, Cmax, and exposure were although under proportional, but we saw increase as the dose increased. And finally, accumulation of anywhere between 1.2 to 1.9 fold was noticed based on day 1 and day 10 PK, so it was sufficient enough to go to the next stage. In terms of safety, we did not see any, it was well tolerated. We did not see any adverse events, no serious adverse events. Some of the adverse events reported were headache and vomiting, and there was no effect on ECG, suggesting that this molecule is safe to move forward to the next level. So when we were running this data, we had preliminary human efficacious dose calculated based on mouse model. But once we got the data from phase 1, we recalculated that, taking into consideration the human PK data. So the PK/PD driver for LXC408 in visceral leishmaniasis is time of 70% of time above EC50 free concentration was sufficient enough. And we also had a AUC over EC50 of more than 300 was sufficient enough to achieve that cure. Based on that, a 60 to 100 milligram was sufficient enough to reach 95% reduction. But in man, we thought of, since it was safe up to 600, we went with 300 milligrams QD, which gives us anywhere between 3 to 5 fold higher window to see whether this compound will be efficacious or not. As I was mentioning, we are in the space of finding out whether this LXC408 direct antiparasitic will have an impact on these diseases. So we are reaching, or as we speak, we are running a phase 2A study in India in collaboration with DNDi. DNDi is leading this study. And we are also planning to go for VL in East Africa. And maybe if everything falls good, we may start a CL study and a Chaga study in Brazil and Latin American regions in the coming years. So it was originated there, and we came all the way here, and with the goal of providing a direct antiparasitic first one in, like with all our research efforts, a oral short course effective therapy which is safe. I didn't want to leave out Chagas, because one of the things which we learned when we came in 2019 is we met Dr. Dilma from Belém region, and she was explaining oral symptomatic acute Chagas disease and desperate need for novel drugs. So I think we are planning to understand how do we make a drug for orally transmitted Chagas disease. We are planning to run a no-fee study, no intervention study to understand that, to come up with new medications. And we also are planning to run study with asymptomatic Chagas disease in this region for our molecules. This is the slide which I wanted to really highlight. It's kudos to all of you who are there and who has put in efforts here. 3 to 4 years back, when I came to Brazil, when we visited for a Latin America drug disease exploration trip, there was nothing in the pipeline for Chagas disease. The top panel was empty except for benzimidazole. And today, 3 to 4 years down the line, we have multiple candidates lined up, at different stages. Kudos to all of you. Hopefully, some of this will reach to the next stage, and we will see some impact, at least an effect of an antiparasitic on the health of Chagas people. Once again, there is a lot overcrowded space in visceral leishmaniasis. I'm just keeping my fingers crossed. At least some of these candidates will be successful, and we can do combination therapy, reduce the treatment duration, and have an impact not only on VL but also CL patients. This has been a tremendous work from many people. I have not been able to capture everybody's name, but here is the couple of teams which are there. And then finally, I just wanted to highlight Novartis, along with the University of Basel, we run Next Generation Scientist program. And there are people from Brazil, Argentina who have participated in that. There is an opportunity for some of your researchers to apply and get that experience of 3 months staying in Basel and learning your interest of area. Now, it doesn't have to be global health. It can be anything which is related to drug discovery. Please apply, and hopefully you will get selected and get to spend some time with us. That's all from me. Thank you very much for listening.
Now we have Dr. Maria Marco. She has already talked a little bit to us yesterday about partnerships, and she's also representing global health projects in the company as a leader in the global health projects at GSK in Spain. Dr. Maria Marco was born in Madrid in Spain and obtained her bachelor's degree in chemistry from the University of Madrid. And after spending a year as a graduate trainee at GSK in Madrid, she moved to London. And I think these are also cool examples for the students to see that there is a possibility that you can spend some time in a company as an intern, and later on that can bring you back, right? And so she has carried out her PhD on the development of novel synthetic methodologies for metal-mediated organic synthesis. And on completing her PhD, she started working at GSK in Madrid, searching for new anti-malaria treatments. In 2011, she moved to the group focused on parasites, where she has been involved in collaborations with DNDi and projects in developing, also in collaboration with the Dundee University, right? Looking for new anti-leishmaniasis and anti-Chagas agents.
Thank you very much for the kind introduction. I'd like to thank the organizers for inviting me today. It's a great pleasure to be here. I'd like to congratulate LOLA and DNDi on their milestone and their achievements so far, and wish them all the best for the next ones to come. So today, I'll start at GSK Global Health. We focus our efforts on 4 main areas: malaria, tuberculosis, antimicrobial resistance, focused on low and middle income countries, and then NTDs and other diseases with focus efforts on dengue and other viral diseases. So the project I'm bringing today is around Chagas disease. And this project was run in collaboration with Dundee University and DNDi. This project originated from one of the heads of the TCACs or kinetoboxes. So this was a set of compounds assembled with the hits that came from the screening of 2 million compounds that are part of the GSK collection. against Trypanosoma brucei, Trypanosoma cruzi, and Leishmania donovani. So in the case of Trypanosoma cruzi, we use the intracellular mastigote form of the parasite, and this compound here, do I have is this, let me see, is there a laser? No. Ah, yes, sorry, I found it, yeah. So this pyrola pyrimidine here was the initial hit, part of the Chagas kinetobox. And this is a moderate hit in the intracellular mastigote assay that uses the virus cell as the host cell. It also had trypomastigote activity and no apparent cytotoxicity from the hepatocytes cell line that we were using, the HepG2. Important to us, we were looking for novel mode of actions, and this compound did not show any activity in the C51 inhibition assay, which is a differentiator compared to posaconazole, which is it targets this mode of action. So also, it's not in here, but I'm for medicinal chemists in the audience. I'm sure you can, with this PIC50 and this kind of structure, you can figure out that it was a good starting point in terms of ligand efficiency as well. So encouraged with that, we move on to a heat to lead expansion effort, where we mostly learn that this pyrola pyrimidine is essential for activity, but also we managed to improve activity just by decorating with minimal substitution around the phenol substituent. So as you can see, the intracellular mastigote activity was greatly improved, maintaining the trypomastigote activity without having cytotoxicity either on the host cell or the hepatocytes. Most interestingly, with this compound, the kinetic solubility was in water was good, and so it was in the fasted is the fasted state intestinal fluid media that we use to mimic the human one, and it was stable both in mouse and human microsomes, and with a low plasma protein binding. So perfect, really optimal ADME profile to progress to our in vivo efficacy studies. So for Chagas disease, we use a different model. Srini mentioned it. It's the bioluminescent model developed by John Kelly and Michael Lewis in the London School of Tropical Medicine. This model has two differentiated phases. The acute phase, where normally occurs 14 days post-infection. And then if you leave the infection to chronify, then you get to the chronic stage normally when you go over 60 days of infection. In this particular case, we were interested in seeing how our compounds would behave in the chronic model. So we started dosing on the 90 post-infection, and then depending on the dose regime, we was either 10 or 21 days. Then you leave the compound to wash out for 10 days and subject the mice for 3 rounds of immunosuppression with cyclophosphamide. And then you leave it for another week before you go for ex vivo organ examination to make sure that there are no parasites hidden in any of the tissues of the mice. So posaconazole fails to cure infection in this model, and this is important to us because we want to avoid clinical failure that was observed with posaconazole. Okay. So when we dose compound 2 at 50 mg per kg twice a day for 10 days, 2 out of the 6 mice show no relapse. And then we thought, okay, well, let's increase the dose or increase the length of the treatment. Still, partial cures were observed. 2 out of 4 and 2 out of 6 mice were non-relapse. In this model posaconazole at 100 mg per kg once a day is fully curative, both at 10 and 20 days. So, and following what Kevin was explaining yesterday, we did monitor that we got exposure during the treatment, and the free exposure of the compound was maintained above the EC50 for the whole duration of the treatment. So that wasn't potentially the cause for this lack of efficacy. So we went on and scratching our heads, thinking what could be the reason for this lack of efficacy. And first of all, we thought about the strain. We are using different strains in the in vivo and the in vitro model. Okay, so we use Silvio for in vitro and CL Brennan for in vivo. However, for all the compounds in the series, we were seeing similar activity against the whole panel of strains. So that wasn't actually the reason causing this discrepancy. We also look into the rate of kill of the compound. So this is compound 2, and all the compounds related within the series are fast and cidal. So that wasn't actually, compared to posaconazole, which is a slower killing rate, okay? And then we thought maybe at the time, there was also in the literature coming out this persistent dormant form of Trypanosoma cruzi was being described. And DNDi was developing an assay that we call the washout assay, where you can actually look at killing all the forms of the parasite. So in this assay, what we do is you treat the parasites with your compound at a very high concentration in a flask. Okay, that's 25 or 50 times your EC50. You treat it for a number of days, and you change the compound every 4 days to make sure that is stable, and then you'll wash out your compound and look for the outgrowth of the parasite. Okay, over a long period of time, because sometimes it does take long. So when we run this experiment with the compound 2, what we did is what we see is when you treat for 5 days at 50 times the EC50, sorry, we were observing that the relapse of the compound was happening at 20, over 20 days. Well, just about 20 days, sorry. It's difficult to see from here, and I don't have all the numbers in my head, sorry. When we did it for longer, for 10 days, we were getting a similar result. Whereas for benznidazole, when you treat it similar, you do not see relapse in the washout. So our conclusion was that we weren't necessarily killing all the parasites, all the forms of the parasite, and that was potentially causing the failure that we were observing in the in vivo model. However, we were thinking that our goal for Chagas disease is to get a new treatment that is safer and or shorter than benznidazole. So what will happen if we were to combine our molecules with the standard of care in a shorter duration? Can we minimize the effects of benznidazole whilst still delivering an efficacious treatment for Chagas? So we thought washout would be the most relevant assay to test this hypothesis. And we went on, and what we did was to combine our compound 2 with a suboptimal dose of benznidazole. So this one relapses quite early. And to a total concentration of 50 times the EC50 to maintain for comparative reasons. And it was at this particular case, we were seeing no relapse after 60 days of treatment. We kept looking in vitro what was characterizing this, and we could actually notice that with our compounds, we have an increased rate of kill compared to benznidazole. So we get a similar rate of kill for benznidazole at much lower concentrations when we co-administer with our compounds. And also we can get a much increased rate of kill when we do that combination. So we thought it was worth testing this in vivo, in the in vitro model, because we could potentially achieve no relapse in combination with the suboptimal dose of benznidazole. So for by then we already had a more advanced compound, this compound 3. Which was slightly more potent. It had similar profile, but better characteristics. And so we took compound 3 and tested it in the same model that I explained earlier. And what we see here, this is in blue, you can see the dosing, duration, immunosuppression, and then after the end of the experiment. Okay, this is images from the mice, the same experiment, and with finishing dosing, and then immunosuppression, and then ex vivo, prior to ex vivo, sorry. So when we treat with compound 3 for 5 days, with benznidazole for 5 days at suboptimal dose. Remember, previously we were using 100 mg per kg, now we're using 30 once a day. Here we do see that only 1 mouse gets no relapse out of the 6 that we were that were in that cohort, that group. Sorry. Then when we treat with our compound 3 on its own for 50 mg per kg twice a day, again, we only see partial relapses. So 3 out of the 6 mice were showing no relapse. However, when you treat, when you took a combination of the 2, so 30 mg per kg of benznidazole once a day plus compound 3, 50 mg per kg twice a day, none of the mice show relapse. So this was a great confirmation of the hypothesis that you can actually achieve a positive outcome when you combine these 2 together. In parallel, this was a phenotypic hit, and what we follow a similar approach to what Srini was describing. So you try to deconvolute the mode of action, and Susan Wiley from the D was successful in identifying BC1 as the target for the spirulopyrimidine series. So unfortunately, oops, that has. Sorry, can we go back? Because I think I've screwed it up. But inhibition of BC1, of the parasite BC1, well, the BC1 complex is conserved across eukaryote. Okay, so that poses a challenge in terms of potential of targets that we can have for the human. And yeah, thank you. So we were interested in seeing the effect that compound 3 will have on the human BC1. So in the enzymatic assay, it does show inhibition of the human BC1. That's not good news for progression. That was confirmed, and calcium loading capacity assay, where we had a minimum effect concentration of 5 micromolar. And again, further confirmed in a mitochondrial stress test or seahorse assay, where we can see a depletion of the oxygen consumption rate. So that was actually stopping this compound for further progression, and we needed to look into finding compounds without this liability. So we put in together a whole screening cascade focused on identifying compounds that had no inhibition against the human BC1 complex, but it would still maintain the TCR BC1 inhibition. And we were successful with this compound 4 here. Again, very potent in vitro with very good, well, with no cytotoxicity, very good physical properties. And in this case, it was inactive in the calcium loading capacity assay and in the seahorse assay up to the maximum concentration tested. So we took this forward to the efficacy study again in combination with benznidazole. This time, again, it was 50 mg per kg BIDs treated during 20 days. And what we get here is, sorry, treated for 5 days, sorry, because we're looking for a shorter dosing regimen, sorry. So when we treat benznidazole 30 mg per kg once a day for a combination with a compound 4, no relapse was observed. And we went on looking to potential for further reducing the dose of our compound. And luckily, we can even go down as low as 10 mg per kg twice a day for compound 4. Still seeing no relapse in the efficacy model. So we have been successful in finding selective and efficacious trypanosoma cruzi BCL1 inhibitor with this pyrrolopimidine series. So luckily, hopefully I've been able to show you that co-administration of the BCL1 inhibitor with benznidazole has potential for 5 days, led to the parasitological cure in mice. This is a BCL1 inhibitor as per the mode of action studies that were carried out by Professor Susan Wiley. And but hopefully, and most importantly, this potential co-administration could be become a therapy for Chagas disease as well in the future. So none of this would have been possible with many people involved with GSK, DNDi, DNDi, London School as well, but also financial support from Wellcome. So I'd like to thank them all for all their efforts and you for your kind attention.
We'll have the microphones passing for questions. And I think we have an online question, which I can wait to receive here. I would like to ask the first question. If you could comment a little more about timeline and costs of drug development nowadays, because we see all these beautiful examples, and often we want to talk about timing and numbers for cost, but we just take them out of somewhere in the internet, right? Because it's really hard to estimate. So I was wondering if you could discuss about that.
Yeah, the question is regarding how do we estimate the cost of developing a drug? That's correct?
Yes, I would say from an initial hit, right?
Yes, initial hit all the way.
To the clinical trials.
I think there are quite a lot of, there are quite a lot of publications available. And I think Jadal was presenting a very nice reference which he referred to. And there you can clearly see that the cost of developing a molecule from initial screen all the way to patients ranges anywhere between $1 to $2 billion. And this includes, actually majority of the cost is coming at the later stage. When you start phase 1, phase 2A trials, phase 3 trials, that's where the costs are very high because human patients are involved and we need to collect a large amount of data. In order to register a molecule, you need to have safety for at least 1,500 patients. Although there are some exceptions like how human African trypanosomiasis, DNDi really convinced health authorities with 300 patient data for fexinidazole. But in generally, it's a long game. We are cheap, discovery place is where we don't spend that much time, but going forward, we have to. And we have done this study internally within Novartis also, and it is expensive. That's how it is, yeah.
Okay, here in the back, Jair Siqueira Neto from UC San Diego. I have a question that might be polemic. So for the Chagas disease animal model, why do you use stereo cure as the endpoint if you think that the patients, 60% or more of the patients live with the parasite without symptoms? So what?
It's a balance as well for, so for Chagas chronic patients, only 30% of them will develop the disease. Okay, so we have to aim for bigger endpoint to justify as well the treatment. So I know the burden is high, but it's also a matter of posaconazole fail in the clinic, and this is the differentiating point in this model. Okay, posaconazole doesn't provide a stereo cure or non-relapses, and this is an additional value that makes us. Remember, it makes us believe that this is progressible. Remember, none of these models have been validated in the clinic yet. Nothing has been progressed since posaconazole, and it's important that the little evidence that we have that we can actually make a difference, we can prove.
You addressed it very well. So the reason why scientific community, especially in the research field, we are heavily relying on relapse-free cure in in vitro assays and relapse-free cure in mouse model is from the lessons what we learned from cytochrome 51 inhibitor, posaconazole. That is behind all our mind. We do not know whether that is the right approach or not, but it is one of the best approaches what we could take given the neglected tropical disease. We cannot come up with multiple compounds. So that's the logic, yeah.
We have a question from the people who are online. Considering the different forms of the parasites, for example, the amastigode and trypomastigodes, how do you choose which form to use for the phenotypic screens? Is it always the infective form, or should you screen in more than one forms?
So in the case of DSK, what we thought what was the most relevant form for the human disease. Okay, so that's why we chose intramacrophage for mastigodes for Leishmania, intracellular mastigodes for Trypanosoma cruzi, and the bloodstream forms for the Trypanosoma cruzi. But that was our approach.
Yeah, at Novartis, especially for Chagas, what we did was we wanted to identify inhibitors for all the different stages. So we started our screen with a full life cycle. So we incubated with compounds right from time of infection with trypomastigodes, allowing the parasites to go inside for amastigodes and to come out of it. So the whole process of screen was slightly 6 days. Parasite life cycle is around 4 to 5 days. That's what we used, initial screen. So we didn't want to miss out on compounds inhibiting trypomastigodes or compounds inhibiting the entry of parasites into the amastigode, or replicating amastigodes, or even developing from replicating amastigodes to that metacyclic form and coming out. So that was the approach we used. But later on what we did was we tried to differentiate the compounds based on specific activities on trypomastigodes, intracellular amastigodes, or even infection ability, whether if you incubate a compound with a trypomastigode, whether this trypomastigode is able to enter and form amastigodes. So these are few assays which we used, but that was not used for initial screening. Initial screening had a large.
And depending on the species, there can always be the challenge that you cannot have an in vitro system with the infective forms, right?
But that's also so far the problem with the persisters. So there's no an assay to test compounds against the persistent form that is amenable for screening compounds for Chagas, for example, and that's important as well.
Hi, I have a question for Maria or anyone who works with Chagas disease maybe. But in your work, Maria, you showed how you systematically screen for CYP51, assuming you don't want compounds who inhibit CYP51. But let's say you work on a specific target, will you still keep screening for CYP51? What happens if you have a dual inhibitor, let's say, and how often do you see CYP51 inhibition in your screenings? Thank you.
Thanks for the question. We, for a specific target, we would only screen if we start seeing deviation in the activity, for example, that we may be picking the C51 as an additional mode of action, unless if you know the mode of the target and it correlates with your activity, there's no need for C51 inhibition. But you start seeing strange things and unexpected activity, and you suspect it could be coming from the C51 inhibition, then we will return to the assay and test the compound.
Hi, my question is regarding the aspect of formulation design, because you show all of the speakers show a very nice result if you consider it since from screening to the clinics. But if you consider a chronic disease like leishmaniasis, for example, mainly for Dr. Howe, what's your opinion about, for example, the possibility to develop control-release formulations for leishmaniasis? And if you, for example, here in Brazil, have some rural areas that the patient compliance is very difficult. For example, with glucanthine, it's very difficult.
So it's a very good question. You know, at Novartis, we are putting a lot of effort on TRD, which is RCPP, what you call compound profiling. So we did have a program just to look at, can we develop a long-acting injectable using our molecules, and what is the dose required in order to have, how long it can be there. So these kind of thoughts are coming in, but once you incorporate any of these ideas, the molecule becomes expensive. And the cost of goods starts going high. As long as we find a relevant paying authorities who can take up some of those costs, then it makes viable sense. But it's a good idea. We are thinking right from the beginning, whether it is implantation, long-acting injectables, or can we make formulation in such a way that it stays longer. But just keep in mind, safety is also very critical, because if it is long-acting and if it is not safe, then we need to come up with a method to block that or eliminate that drug very quickly. There are some challenges, but it is going in the right direction. Thank you.
Thank you. Jean-Jose, so from DNDi, so great session and really nice talk for the four of you. I have a question for Mike on flubantilosine. Obviously, we're equally disappointed about the results, such a great story and such a great compounds that you put through the development. You formulated a number of hypotheses for the failure on efficacy. Are you also considering possibly some sanctuary sites for the parasite to hide, and that possibly that drugs due to suboptimal permeability, tissue permeability, could actually not reach? Is it something that is an hypothesis at all?
Yeah, I think that's a possibility. I don't think we have any data to really speak to that. Yeah, it's very disappointing. You know, there's really no smoking gun when we look at the data from the trial. everything seemed to line up with what it should have been. If we had seen, the result was pretty clear cut. There was really clearly no efficacy at all. If we had seen some efficacy, then we would have said, well, maybe if we had been able to go to a little bit higher dose or a little bit longer duration or something, we may have seen something. I think the thing I wish we knew is if they had collected samples at the end of the dosing period to see what the drug levels were at that point in the worms. But we don't have that data. They waited till 6 months to take for the efficacy readout.
I have one more question here too. I was thinking about the artificial intelligence tools, right? When you add them to the pipeline, again, still thinking about timing and costs. Do you have experience if it's speeding up the process and guiding to chemical structures, right, that are more suitable for candidates?
Yeah, I'll speak of our limited experience. So the models that were put in place from two different groups, what that allowed is to pre-filter the library. So a library that normally will give you less than 1% hit rate, all of a sudden the filtering allowed for a 15 to 25% rate of positives. That is very significant, and that allows you to remove up front a lot of things that are likely to. And also working with our collaborators in Argentina, for example, that also makes that very feasible from a cost point of view. It's a lot of human time, but less of laboratory and reagent time for consumables, and that is. I mean, the issue with the consumables, at least in Argentina, is of course the price. That is a differential price for Latin America of the various reagents, but also the timing to receive. Well, I don't need to tell you about that. That's an issue.
Okay, Lydia, very inspiring talks. Thank you so much for sharing your time and experience with us. And I have a question for Michael and another general one. Michael, have you shared with us the hypothesis to explain why your compound failed? I'd like to know if you figure out the possibility that you working with an active metabolite, if it has been considering in his work. And the general question is, yesterday we studied a lot about the importance of the distribution of the drug to the target organ. In case of Chagas disease, I think it's most important. Do you think that it's possible to introduce this kind of approach in your works?
Okay, I guess first, is there an active metabolite? I guess I don't. I'm sure that was looked at. To be fully transparent, I didn't work on this project. So I don't think that's the case, but I guess I don't know off the top of my head. Sorry about that.
I think the second question for both of us, it is related to, currently what we are measuring is plasma PK or blood PK. Given Leishmania and Chagas, they have the ability to reach multiple different tissues. As you saw from yesterday's Kevin Reed's presentation, we are trying to adopt what is the free concentration, whether the drug is able to reach these highly either vascularized or non-vascularized tissues. To a certain extent, what we adopted at Novartis, given limited resources, is that we were heavily relying on KPU use, or volume of distribution reaching these different tissues as a surrogate for reaching different tissues. And at the same time, we did not run this for most of the compounds, but for few compounds, we did a whole body MS in rat tissues. So what we do is we give rat or mouse 100 milligrams, 30 milligrams, whatever dose, and we do a mass spec based assay by cutting open the complete tissue, like rat or mouse, and by mass spec we were able to analyze. For our benefit, the series, whichever we worked on, had a kind of a nice distribution across. Although we couldn't do with LXC408 because it was already in the clinic. The other molecules, like slightly, runners which were earlier, they had a better distribution. The challenge of tissue distribution is free concentration. When you measure tissue binding, you may not get accurate results. So that is one of the biggest challenges. We are trying to look at it, especially when we were working on sleeping sickness. We were trying to collect brain, and we were also trying to collect CSF fluid to measure what is the concentration of drug which has gone from blood, which has crossed blood-brain barrier. CSF fluid was excellent marker. Brain, we had an issue with respect to brain tissue binding. But although we used that as a surrogate in order to work for that program, but it's a good question. Thank you.
Yes, to add, reemphasize, because we're using the approach that Professor Reid explained yesterday, the drug efficiency index, as in vitro measurement of potential distribution of your compounds in vivo.
We can take one last question.
Hi, my question is for Serene. Serene, considering the initial screenings that lead to the discovery of the proteasome inhibitors, there were natural products among those libraries? And if there were, how was the hit rate for the natural products?
Yeah, that's a good question. I think if I recollect, we had approximately 25 to 30,000 natural compound library in that particular big screen. We did hit upon a couple of inhibitors which inhibited trypanosoma brucei and cruzi. We did not prioritize them because when the chemists looked at those structures, they felt they may not be amenable for medicinal chemistry optimization. So they were deprioritized. And some of these were also not hitting, they were not in that central core of active across 3 parasites. Few were specific to T. cruzi, few were specific to T. brucei and Leishmania. And since our aim was to identify pan-kinetoplastid inhibitors, we focused on those 85 to 100 hits. Yeah, thank you. Yeah.
Okay, we'll have a break and come back at 4:30. Let's thank the speakers. Thank you so much for being here.